Hello and welcome to our biochemistry review session. Today we will be talking about the amino acids, proteins, protein purification methods, and various high-yield topics in genetic expression. So let's begin by first examining this very important table that divides for us our amino acids into four major groups, actually two groups, but the second group is divided further into polar neutral, polar charged, oppositely charged, such as these histidines, the lysines, and the arginines. These are the basic. So you're either polar neutral. These guys are amino acids who are polar. They can dissolve in water, but they are neutral. They have no charge. But here, green and purple, they are also polar, but they are polar and charged. And we know there are two charges. There's a positive charge and a negative charge. So if you are polar, positively charged, you are lysine, arginine, and histidine, and you're always having a positive charge, a positive charge. And if you are polar and negatively charged, if you are polar and negatively charged, that means your carboxylic group is ionized and it is negatively charged. In the positively charged polar groups, the amino group is ionized and it is positively charged. And this is happening at physiological pH, So the nonpolar groups are very easy to distinguish because they do not dissolve in water. They do not dissolve in water and they are never polar and they obviously are not charged. It is the polar groups that require you to first identify that they are polar, having an OH or an NH and having a charge as such. But we will get in detail. Let's first not forget that the structure of the amino acid is always beginning with an alpha carbon in the middle. Alpha carbon, alpha carbon, alpha carbon, alpha carbon, alpha carbon. This alpha carbon must have four terminals. Three of the four terminals on every single amino acid are always identical. No matter if we're looking at a non-polar amino acid, polar neutral, or polar positively charged, polar negatively charged, we will always have three out of the four identical. And the three are on the left, always on the left, and in the beginning. You know, we write from left to right. Even if you're a lefty or a righty, you use your right hand or left hand, but still you have to write from left to right. So always the peptide chain begins from the left and from the amino terminal. So if you have a polypeptide that is named glycine, alanine, valine, cysteine, and proline, and they ask you on the exam, which one is the first amino acid or which one is on the most left, then you know that that's glycine. That's the first one. Which one is the last one? That's proline because it's all the way to the right. And to the right is always the carboxylic. Carboxylic. It has a carbonyl group attached to an OH or an O minus. And we will talk more about that. So, you know, we have four terminals, three of them are always the same in every single amino acid in nature. And on the left, you have the left terminal, that's your amino terminal. You see amino terminal, amino terminal, amino terminal, amino terminal. On the right, you have carboxylic terminal, carboxylic terminal, carboxylic terminal, carboxylic terminal, carboxylic terminal everywhere. And those two terminals are very important because if I want to connect my amino acids, now they are micromolecules. They are micro, they are a single. If I want to join them and convert them into macromolecules, then I have to make a bond. And that bond is made between the carboxylic terminal of amino acid number one, in this case glycine, and the amino terminal of amino acid number two, in this case alanine. And we talked in class about how it is always the carboxylic terminal donates an OH, and the amino terminal donates one H, and you have O and two H's, and water leaves. That is known as dehydration. And then you make that very important vital bond known as the peptide bond. And then we will make a long chain, and that chain is always made of peptide bonds. So we make the peptide bond by removing water, dehydration, devoiding water. And if we want to separate the bond, we want to cleave the bond, we want to lyse, break the bond, we use water lysis known as hydrolysis. So we add water, and you have to know how is the water going to be added. So the water is oxygen with two H's. So OH goes to the carboxylic terminal on the right of the first amino acid, and that leftover H goes to the other amino terminal of the second amino acid, and now you broke the bond. And we will talk about that. Please remember, this is very tricky because cysteine, and if you examine cysteine and methionine, you see cysteine and methionine, both of them have this thiol group, okay? But cysteine is able to bind. So you see the sulfur group, it can have two bonds. So in cysteine, the sulfur has a bond with this carbon, and the carbon has a bond with this carbonyl, and it can form a disulfide bridge with another cysteine. So if we have another cysteine, this H is going to get removed, and then that S is going to bind with the S of the other cysteine. And that how we're going to be able to make a disulfide bridge. But let's go back to our amino acid structure. That's our alpha carbon, alpha carbon, alpha carbon, alpha carbon, alpha carbon. You have your, to the left in the beginning, your amino terminal, amino terminal, amino terminal, amino terminal, amino terminal. And to the right, you have carboxylic terminal, carboxylic terminal, carboxylic terminal, carboxylic terminal. And to the north, you always have hydrogen, 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 hydrogen. Those are the three out of the four terminals that are always identical between all the amino acids. It is only glycine that has the R group, this box, this shaded group here in orange, here in blue, here in purple, here in green. That's the R group. This R group is very important because that R group is the group that gives the identity to the amino acid. If my amino acid is nonpolar, it is because the R group is nonpolar. If my amino acid is polar, neutral, it is because my R group is polar and neutral, has no charge. If my amino acid is polar, positively charged, it is because my R group is polar and positively charged with the nitrogen. You cannot be positively charged with oxygen. You're always positively charged with nitrogen, or you're negatively charged, as in here, with oxygen. So you must be able to distinguish your alpha carbon and the terminals of the alpha carbon. To the left of it is your alpha amino because it's connected to the alpha carbon. To the right, you have your carboxylic terminal, but we call it the alpha carbonyl. This is the alpha carbonyl. And the R group is what gives the amino acid the identity, whether or not it is polar or non-polar. We talked about that. Okay, so glycine is very interesting because its R group so happens to be a hydrogen, just like its hydrogen terminal. So it has two hydrogen terminals. One of them is the main hydrogen terminal. The second one is the R group, who's also hydrogen. And that what makes glycine lose the property of being chiral. In order to be chiral, all the four terminals have to be different. Like here, hydrogen, carboxylic group, methane, and amino group. That's a perfect example of a chiral. That's a chiral amino acid. That's a chiral amino acid. You see all of the groups are different. You can examine them for yourself. All the R group is different from the amino terminal, is different from the hydrogen, and different from the carboxylic terminal. So you must see that glycine there. I think there's one question on the exam that says which of all the amino acids is achiral or not chiral, and that would be glycine. So just please make sure that you take a good look at this table here with the amino acids, and you must be able to distinguish your leftmost group, that's the amino, and your rightmost, that's the carboxylic group. Then that is the hydrogen terminal, and this is your R group. The positively, then that's one category, non-polar. Polar then is divided into three groups. There is the polar that is neutral, doesn't have a positive or a negative charge, or you can have polar that is positively charged. And whenever we say positively charged, you know it's positively charged because of the amino group. And these are your lysine. You see it has an amino with a positive charge. Arginine, an amino with a positive charge. Histidine, amino with a positive charge. And it has a positive charge because the amine group is usually has NH2, two hydrogens, and a lone pair. But when that lone pair is taken, and instead of it, that amino group has a third bond, it will now have a hydrogen, an additional hydrogen. It will become protonated. Protonated. It has now an extra hydrogen, and that gives it a positive charge. That is the opposite of oxygen, because oxygen gets deprotonated, deprotonated. So this OH is no longer going to become an OH. The H is going to be lost. And in exchange for the H, the oxygen is going to get an electron pair. Okay, it's going to get a minus charge. And that is how the carboxylic terminal is deprotonated. And we talked in class about how this amino acid is actually a buffer. So you see here this NH3 and this OH minus. What happens is when your body is suffering from acidosis, there's so much acid, the amino terminal will say, I am a proton acceptor, I'm a hydrogen acceptor. Give me those extra hydrogen proton, because you know acidity means low pH. The pH is so low, and it also means that the hydrogen proton concentration is so high. So the amino terminal will say, I'm a buffer, I can come to your help. You're suffering from acidosis, you have so much hydrogen proton concentration, give me some of those. I'll take the burden away from your blood, and it will work like a buffer. Then the other side, the carboxylic terminal will do the opposite. It has an H, it has an OH here. So when you're suffering from alkalosis, alkalosis means high pH and very low hydrogen proton concentration. So the OH will say, oh, I have an extra hydrogen, you can have it. And then it will start supplying you with hydrogen, supplying your blood with hydrogen proton concentration. So that's what makes it act like an acid. So the carboxylic terminal is an acid and the amino terminal is a base. So our polar groups are neutral if they are in a state of OH, NH, like here, NH2, OH. These are polar neutral. They didn't write it here, but it is polar neutral. OH, NH, SH, that's fine to SH, that's polar. So for example, this group here, they usually put it with non-polar because it can form a disulfide bridge, but it is a polar amino acid. And the groups that are polar, but they are acidic, they always have a charge that is a negative charge if you are an acid. And you know why it's negative? Because it lost the OH, it lost the H that was bounded to the OH. It lost the H and got an electron instead in its place. And here, if it's positively charged, that is because it gained an additional hydrogen proton. Because remember, it's a base. It accepts hydrogen proton and a positive charge. And the acid donates hydrogen proton and gets a negative charge. So that's why aspartic acid and glutamic acid are negatively charged, because they are the ones who supply. They get deprotonated. They remove the hydrogen proton, the H, the hydrogen proton gets deprotonated, gets removed, and in exchange, they get paid with a minus electron. So they donated the hydrogen proton. These are acidic because they donate the hydrogen proton. And these guys, they are basic. They accept hydrogen proton. And as a result, they gain a positive charge. Okay. So the basics of the amino acid structure, as I said, is you have your alpha carbon. You have your alpha carbon. We can restructure it differently, but here to help us analyze it some more, that's our alpha carbon. The alpha carbon has a carboxylic group with an OH. You see the carboxylic group. It has an OH, and that's a carbonyl. That's an alpha carbonyl because it's close to the alpha carbon. That's my alpha carbon, and that is alpha carbonyl. And this is the alpha carbon right next to this amino group. So this amino group is called the alpha amino. So this is your central alpha carbon. Next to it is your alpha carbonyl. And this whole terminal is the carboxylic terminal with an OH. That's your acidic terminal. That's the terminal ready to give you a hydrogen proton when you need it. Like when you are suffering from alkalosis, you don't have enough hydrogen proton. This terminal will say, here you go, you can have my H. And this is amino terminal that has two H's and it's ready to accept more. So if you're suffering from acidosis, too much hydrogen proton, she will say, I can take from the burden that you have. You have so many hydrogen protons. Your pH is so low, you have acidosis. Give me the hydrogen proton. And this is the third terminal, the H, and that is the R terminal that gives the amino acid its identity. Okay, so this is the alpha amine, that's the alpha carbon. We talked about that's the alpha carbon, and that's the alpha carboxyl group, and that's the alpha amine. Okay, so the differences in our group example are, so for example, look at here, you must be able to look at this diagram. So this is a wedge. A wedge just shows you that this is a stereoscopic view, that this NH group is coming at you in the model, and these groups are in the back. So here, where is my alpha carbon? Well, that's very easy because I can see my carbonyl. I can see my carbonyl group. My carbonyl group is right here, and that's my OH, so that's my carboxylic terminal. It's my carboxylic terminal. And right next to my carboxylic terminal, this dent here represents for me a carbon, represents for me a carbon. And this carbon is very is attached to the alpha amino. So that's my alpha amino. So that's my alpha carbon. That's my alpha carbonyl. That's my alpha amino. And the hydrogen group is not shown because we don't show the hydrogen group in this particular type of model. So here there's two hydrogen groups, but we don't show them. We do not show them, but we know that they exist. We only show the hydrogen if it's bounded to something other than carbon. So for example, here, that's a carbon. It has 4 bonds. Here, that's the alpha carbon. That's the alpha carbon. It has a bond with the carboxylic terminal. It has a bond with the amino terminal. And it has a bond with the hydrogen, but it's not showing. And it has a bond with the R group. So that's my R group. So here, very interesting. Who's my R group? My R group that's my alpha carbonyl. That's a carbon. That's an SH. Who does that look like? Let's go back into so that's a alpha carbon with a carboxylic terminal, amino terminal, hydrogen, a carbon, then an SH. That's cysteine. So that was cysteine. This is cysteine. See, that's cysteine. That's the carbon in the middle, then that's the cysteine. This is the formation. This is the amino acid that is able to participate to make for us disulfide bridge. And these disulfide bridges are important in stabilizing tertiary structures in the protein folding. So the protein folds from primary structure to secondary structure to tertiary structure, and these cysteine disulfide bridges are very useful into the folding process. And they get denatured with an agent known as mercaptoethanol, and we will see that. We will see that in a little. So that's my R group. That's my R group. And this is my alpha amine, and that's my alpha carbon, and that's my alpha carboxylic group. And this shows an asymmetric center, chiral center. You see, asymmetric meaning that it is chiral. I'm sorry. Asymmetric means it's chiral. So all the 4 different sides are different in their structure. So that's a carboxylic group, that's an amino group, that's the R group, that's a hydrogen group. Okay, what's this big circular group? That is known as an aromatic group, an aromatic group. So now we have replaced the R here. Here our R group was a thiol group and the entire structure was cysteine. But here our R group is an aromatic group. And let's go and look at who looks like that with an, that is phenylalanine. You see, phenylalanine has this aromatic group. So the only difference between cysteine and phenylalanine is that cysteine on its terminal group in the R group is composed of this carbon that is attached to the alpha carbon and a thiol group, the sulfur. But here, instead of the thiol, the sulfur, you have this very important aromatic group, and that is the phenylalanine. That is a non-polar amino acid. Okay. You see, that is our alpha carbon. That's our alpha carboxylic group. And that is our alpha amine. And this is our R group. So isomers show us a different configuration with the same exact molecular structure. So being an isomer means that you have the same number of carbons, the same number of hydrogens, but you are arranged differently. We will see this when we discuss carbohydrates. We will see that glucose, fructose, and galactose, they all have the same number of carbon and hydrogens, but the way that they are connected, the way that these carbons and hydrogens are connected, they are not connected in the same way. So when we eat foods rich in glucose, the glucose is going to get metabolized in our cells to give us ATP by entering a pathway that is known as the glycolysis. But when fructose and galactose get into our cells cannot shuttle or shunt this fructose and galactose into the glycolysis because glycolysis can only accept glucose. So there are enzymes known as interconvertase. Interconvertase enzymes, they convert fructose into its isomer glucose. They convert galactose into its isomer glucose, and then they can enter into the glycolytic pathway to give us energy. So when you drink milk, which is rich in glucose, when you eat fruits, I'm sorry, milk is rich in galactose. When you eat fruits that are rich in fructose, these are not glucose. When you eat sugarcane, that is all glucose. When you eat grain sugar, when you eat grain sugar or oatmeal, that is all glucose. It goes immediately into the glycolytic pathway in glycolysis to give you ATP. But when you drink milk, that galactose has to be converted into its isomer glucose, then we can get ATP out of that. That's why people who are lactose intolerant, they lost that in enzyme. They lost that enzyme that is able to convert this isomer into its glucose isomer. So here that shows you we have an asymmetric carbon, but the way the structure, they have the same number of carbons, but the way they are arranged in space are different. Okay, let's look at how the peptide bond is formed. That is very important. In the peptide bond, we said that if this on the leftmost, that's the amino terminal, known as the N terminus, and on the rightmost, that is the C terminus. So each amino acid has an N terminus and a C terminus, has an N terminus and a C terminus. When you connect them, you will have a shared N terminus all the way to the left and a shared C terminus all the way to the right, because now they are connected like a train. The train carts, they do not have, they're no longer separated. They are connected by a peptide bond, and the head of the first amino, but the head of the first cart is going to be the head of the train, and the tail of the last car is going to be the tail of the entire train. So now let's look at what happened. That's amino acid number one. That's amino acid number two. If I want to connect them together, I make this green bond right here. So how did I go from R1, who's separate from R2? On my C terminus, why did they call it the C terminus? Because it's the carboxylic terminus. It is all the way to the right. You see, that's the C terminus. It has carboxylic acid. C terminal, that's a carboxylic terminal. And why did they call this the N terminus? Because it is all the way to the left. That is the N terminal. All the way to the left, that's the N terminal. So now in order for them to be bonded, to be connected, what happens is the C terminus will donate an OH because it's carboxylic acid. It will donate an OH, while the N terminal, that is the base, the amino terminal, will donate one H. And you will have the sum of O and two H's, that's water. And then water will be removed and water will be taken out. And then we will connect this bond. And if you want to cleave, so now you have a dipeptide. So you have because it's made of two amino acids, that is known as a dipeptide, two of them. Okay. So these are the peptide bonds. You see, that's the N terminus of amino acid 1. That's the C terminus of amino acid 1. That's the N terminus of amino acid 2. That's the C terminus of amino acid 2. In order to connect, remember that it is always the C terminus of the first amino acid. It will not give me one H. It will give me an OH. And it is always the N terminus of the second amino acid. It will not give me two H's. It will give me just the one H. So I can have a sum of two H's and O, and that's water. And then all of that will be removed in order to give me water. So that's the alpha carboxylic group. That's the alpha amino group of the second amino acid. And what happens is they get attached together. Like that's a dipeptide. If this happens somewhere else, that will be, if we add another amino acid, it's a tripeptide. So now they increase the number. But remember, it's called dehydration because you see, you began with that, and then enzymes catalyze the removal of water, the dehydration. So now you have the bond connected, and don't forget your water that comes out also in the product. So that's your product. When you connect the two, you had two amino acids. Now you have a dipeptide as a product with a peptide bond, and don't forget your water that came out. And that's why they call it dehydration, because the water came out. So the primary protein structure is always amino acids like that. So see, those two, dipeptide, then tripeptide, they get connected one after the other until you have a long string of beads. And that is the structure, the primary structure. That is the most important structure. And we discovered that, what Christian Anfinsen did in his experiment that led him to have the Nobel Prize is that when the protein denature. What do I mean by denature? Denature means you break down. You break down from a quaternary structure to a tertiary structure to a secondary structure, and you end up at the primary structure. Denaturing means we break the bond that led us to fold. So what bonds led us to fold? When we are in a stage of primary, what bonds are there? It's these peptide bonds. These are very strong bonds. But when we fold upon ourselves to go from primary to secondary, then these are no longer peptide bonds. These are hydrogen bonds. So more hydrogen bonds, more extensive hydrogen bonding happens that leads us to fold from primary structure to secondary structure, which are alpha helix and beta sheets. Then more extensive hydrogen bonding and hydrophobic effects that and disulfide bridges and salt bridges that gives us tertiary structure like myoglobin. Then adding ionic bonding to the top of all of these hydrogen bonds and hydrophobic effects and van der Waals and the salt bridges and the disulfide bridges. That gives us a quaternary structure like hemoglobin. So when you heat up these quaternary structures, these tertiary structures, you heat them up, you change the pH, you change the solubility, then all of these weak bonds, the ionic bond, the hydrophobic effect bonds, van der Waals bonds, the hydrogen bonds, all of them, and the disulfide bridges, they fall apart, but the peptide bond is never affected. So then if you restore the optimal conditions, then these amino acids start to aggregate and fold upon themselves again. So he discovered that it is the most important structure is the primary structure of our protein sequence. That's the most important. But remember, just because I have additional hydrogen bonding, just because I have additional disulfide bridges and hydrophobic effects and ionic bonding, it doesn't mean that the peptide bond went away. The peptide bonds are always there. They are the ones who make my train, my primary structure. But those other weak bonds, they are additional bond to help me fold and perform conformational folding. So when my protein denatures, that means we only lose the weak bonds. The peptide bond will never break. If you break the peptide bond, you destroy the primary sequence, and then you can never refold again. So the primary protein structure is made up of linear sequence of amino acids. They are joined by peptide bonds, and they are translated from RNA using, we talked about that, using that translates from RNA in the ribosome using the genetic code with the codons and anticodons. And the synthesis begins at the amino end and terminates at the carboxylic end. That's why the amino is at the leftmost, it's the first one, and the carboxylic is the rightmost. Ultimately determines all the properties of the protein. So this is the primary sequence that determines all the properties of the protein. How is it going to fold? Is it going to have a disulfide bridge? Is it going to be is this protein going to be acidic? Is it going to be basic? Is it going to be an enzyme? Is it going to be ionic channel? So that is determined by the primary structure. So this is also very interesting because we see now we are training to, this is a polypeptide. It was formed by dehydration. So now let's find our peptide bond. You see our peptide bond is always between the carboxylic terminal with this alpha carbonyl. That's my alpha carbon. That's my alpha carbonyl. On the other side, that's my alpha carbon, and that's my alpha amino, and they connect like that. So this is glycine because you see it has two R groups. One of them is an H, and the second R, sorry, it has two groups. One of them is a hydrogen, and the second R group also happens to be coincidentally hydrogen. So it has two hydrogen, and it's achiral. It does not, it has a symmetry because it has two hydrogens. So that is the first peptide bond. And now let's look at if we have a second, you see that the second peptide bond, that's my second alpha carbon. And then that's my third peptide bond. And that's my third amino acid. And that's my fourth peptide bond. And that is my fifth peptide bond. So the peptide bond is always, the number of the peptide bonds is always the number of the amino acids minus one. So I have 1, 2, 3, 4, 5, 6 amino acids minus one. That is 4 peptide bonds. Let's see it. So one peptide bond, two peptide bond, two peptide bonds, three peptide bonds, and four peptide bonds, four peptide bonds, and one peptide bond, two peptide bonds, three peptide bonds, four peptide bonds, and five peptide bonds. So the peptide bonds are always the number of the amino acids minus one because you connect 6 amino acids with 5 peptide bonds. That's the amino terminus, and that's my carboxylic terminus. That's the free carboxyl group. And that's the free amine group because it's at the end of my polypeptide. And that's the end of that's the beginning of my polypeptide. And that's the end of my polypeptide. That's the leftmost, that's the rightmost. These are the peptide bonds. You see, peptide bonds. And these are alternating orientations of the R groups. So they can change their position in space. Okay, the secondary structure is the alpha helix. The alpha helix is when the polypeptide, you see this polypeptide starts folding upon itself due to, you see the hydrogen bonding, this nitrogen and the hydrogen, they have a polar covalent. Remember, we talked about that in class. And this here is a covalent bond. So now the hydrogen, who is more electropositive, will start to have this dotted weak bond known as the hydrogen bond. So now this hydrogen bonding will lead the primary structure to fold upon itself in a helix like that. See the hydrogen bonds, they stabilize the alpha helix. There is another type of secondary structure known as the beta sheet, and it's also based on hydrogen bond. You see the hydrogen bond is always between the hydrogen of the nitrogen and the oxygen of this other amino acid. The hydrogen of the nitrogen of one amino acid and the oxygen of the second amino acid. And they are, sometimes you can have them parallel, sometimes they can be anti-parallel. This is the beta pleated sheets, the beta pleated sheets. And these are the hydrogen bonds between the hydrogen and the electronegative oxygen, because this hydrogen is more electropositive because it's in a polar covalent bond with this nitrogen group. Okay, that's the structure. You see, now they are, here they are anti-parallel. They're anti-parallel. They are in opposite directions, like here. They're anti, but you don't have to worry about that. Just remember that these uh, secondary structures come either in the form of alpha helix or beta pleated sheets. That shows you an example of the usefulness of proteins that are in secondary structure, like collagen. You know, collagen is the most abundant protein in the body, and it is in a secondary structure. How interesting. Keratin and fibroin that is found in silk, keratin found in the hair and nails, collagen found in the ligaments of the bone. These are very important proteins, and they are good enough just at the secondary structure. They don't even have to become tertiary, like myoglobin. Then the tertiary structure, that's when you have even more extensive hydrogen bonding, disulfide bridges, and sulfide bridges, and hydrophobic effects. You see, the tertiary structure is made up of alpha helices and beta sheets that are joined together by these hydrogen bonds and these salt bridges and disulfide bridges. And then this shows you an unfolded protein in a primary structure, and then this unfolded protein will undergo hydrogen bonding and disulfide bridges and hydrophobic effects, and then ion bonding to become folded. Or it can unfold. You see how it's folded in many different subunits. When we heat it up or change the pH or change the salinity, what happens is only the weak bonds, which are the ionic bonds, the disulfide bridges, the salt bridges, the hydrogen bonds, and the van der Waals effects and the hydrophobic effect, they all break. And you just end up with this primary structure. But the peptide bonds are never affected. So please remember that denaturing just refers to us going from quaternary or tertiary, secondary back to primary, but we cannot go below primary. That is not called denaturing. If you destroy the primary structure, then that's not denaturing. That's destroying the entire blueprint. That manuscript is gone now. But denaturing refers to just the destruction of the hydrogen bond and the salt bridges and all of the using some detergents and using some agents that break the salt bridges and break apart the disulfide bonds and breaking the hydrogen bond with temperature. So now we're just breaking these weak bonds. That is denaturation. Denaturation is breaking the weak bonds and unfolding from quaternary to tertiary to secondary to primary. But if you break the peptide bond, then that's not denaturation because temperature cannot break that bond unless it's severe temperature. So denaturation refers only to the weak bonds that are broken. What are the forces? What are the forces stabilizing tertiary structure? Hydrogen bonds, we talked about that, hydrogen bonds, and you see the hydrogen bonds stabilizing the tertiary structure. You see how the hydrogen bonds are? These are the dotted bond between the electronegative oxygen and the electropositive hydrogen. Electronegative oxygen, electropositive hydrogen. These are weak bonds. These are bonds that can be destroyed with pH changes and the temperature changes and so on. Disulfide bridges are covalent bonds. So these disulfide bridges are when you see that's cysteine. That's cysteine. What happens is the two hydrogens get deprotonated. The two are removed. You see the two are removed, and in their place, the sulfur the thiol group, the sulfur gets an electron, and then a bond is made. So the two hydrogens are gone, and then a connection. That's the bridge. That's the disulfide. It's called disulfide bridge. It's connecting two sulfur groups. You see, these are now two amino acids that are cysteine connected by this sulfide bridge. That's important for tertiary structure. This covalent bond, it is very important in the tertiary structure, and it is broken by a very special detergent known as mercaptoethanol. We will see that. Okay, denaturing and unfolding. So the denaturing and unfolding of protein is breaking forces that are stabilizing these globular proteins. Like here, mercaptoethanol or diethyltriol, these break the disulfide bridges. So you must remember that how do I break this disulfide bond? It's by using this detergent known as mercaptoethanol. This is the only one that breaks the disulfide bridges. So if I want to denature a tertiary structure that has disulfide bridges, all I need to do is add mercaptoethanol. It's going to cleave this bond right here. And that's going to denature my protein. Detergents are important for disrupting the hydrophobic reactions that help stabilize the folding of these proteins. Heat and pH also, they are very important because they break the hydrogen bonds and they change the charge and alter the ionic interactions. And chelators, they are important because they are agents that bind the metal ion. The metal ions are part of the protein R group, and they are important. The metal ions are important for stabilizing the folding of the protein. So if you use a chelator, you're gonna bind the metal ions and cause them to break apart and not hold or staple together these folded proteins. So that's the primary structure. And if I want to fold it, you see the folding with the disulfide bridge is always going to happen between the cysteine groups. And you see that's a mature protein tertiary structure. There are consecutive intra-chain disulfide bond. You see this intra-chain, within the chain, intra-chain. And there are also non-consecutive intra-chain disulfide bond. You see, so now one represents consecutive. So they are right after the other. So you see this cysteine group. So that's the beginning, the cysteine, then the nearest cysteine and that cysteine are connected. This is known as consecutive intra-chain disulfide bridge. Here also, this group is followed by this amino acid. This cysteine right here is also attaching with this cysteine who's right next to it in order. But if you look at these two cysteines, they are far apart from each other. They are not consecutive. So you have this cysteine who is not bonding to this cysteine or that cysteine. It is bonded to, it skipped two cysteines. So this cysteine skipped this one and bounded to the third one. So these are non-consecutive intra-chain disulfide bridges. But all you need to know is that it is mercaptoethanol. That's the one who's going to break these disulfide bridges. And you know the disulfide bridges are important for going from primary, folding our way through to become tertiary. So when you use mercaptoethanol, we're going to break that bond and we're going to return to primary structure. Okay, you see this is my cysteine. Look at the structure of cysteine, and through oxidation, the two hydrogens are going to be removed and you're going to make a disulfide bond. Then if you use mercaptoethanol, you're going to go in the opposite direction and you're going to break the bond and you're going to return back to two cysteines. Okay, so the denaturing is, so that's my folded protein. If I want to go down this direction, I add urea and mercaptoethanol, and they break these disulfide bridges and salt bridges, and you will have a long folded chain. But this is a primary sequence. But if I want to fold up, I remove urea and I remove mercaptoethanol. And what's going to happen is I'm going to refold right back up into a tertiary structure. Okay. The primary structure is made up of an amino acid sequence. The secondary structure or the super secondary structure is made up of repeating structures. And the tertiary structure is made up of folded structures that are longer in range. And now we're going to talk about how we separate proteins. So in order for us to decipher what is a protein made up of, who are the amino acids, what is the nature of the R groups, we have to break into the cell, get the solution that has all of the proteins, and now we have to learn how do we figure out and analyze the identity of that protein. So we have to purify the protein. So the first thing is we centrifuge the solution. And when we centrifuge the solution, we end up with an entire fraction that is only protein. So we use our pipette and we remove that layer that has only the proteins. And now there are different ways for us to purify our protein and figure out purifying it, meaning separating its structure so we can see what it's made up of. Okay, so like I said, in order to this is not purification. That's our way to start the purification. We must first open up the cell, cleave the cell membrane, get the content of the cell, the cytoplasm, because the proteins are all in the cytoplasm. Okay. And then we centrifuge, and all the stuff that is not relevant to proteins is going to be in the supernatant. It's going to be all the way at the top. We don't need that. That's useless. And we always going to go to the bottom. That's the pellet. And we're going to take all of the proteins that are sedimenting and they are so dense at the bottom. Then we're going to collect all of them. And then we separate them based on special ways. So the first way is chromatography. Chromatography is very important because it separates between proteins based on different properties. So for example, there's a chromatography that separates proteins based on the charge. So it uses a protein medium that is negatively charged. And when you pour your protein solution, the negatively charged amino acid residues are going to get repelled because our medium is negatively charged. So the negatively charged are going to get repelled, and the positively charged are going to stick. They're going to stick to our medium. So now I separated the positively charged amino acids from the negatively charged amino acid by using a negatively charged medium that would stick based on the charge. So here we separated our positively charged from the negatively charged by using a filter or by using a medium that is opposite to what we want. If we want positively charged proteins, our medium is going to be negatively charged. So when we pour our solution, only the positively charged will stick to the negatively charged medium. But if we want negatively charged amino acids, our medium has to be positively charged. So the positively charged medium is going to, like a magnet, absorb all of the negatively charged amino acids, and the positively charged are going to be sent out. There's also separation based on the size. So we're going to use gel filtration where there are beads, and the beads, there's many of them, and the beads have little holes inside them. And you pour your protein solution. The really tiny proteins that you do not want are going to get to the bottom so slow because they have to travel through the beads, not just between the beads, but through them. But the protein that you want is going to be so big, it cannot go through the beads. It will just zigzag through the gaps, and that's how you're going to collect it. We will talk more about that. There's also separation that is based on the affinity. Separation that is based on the affinity. Like, for example, remember in the DNA, we said, the RNA polymerase is going to bind to the structure of the DNA, and for every G, it's going to have a C. For every C, it's going to have a G. So something like that. Our medium now is not going to be having a charge like in the ion exchange. Here it's going to have like a DNA sequence, or maybe an antibody, antigen, or maybe an enzyme and a substrate. Two things that will bind with affinity like a lock and a key. And then when you pour your solution, then your medium is going to be, for example, an enzyme, and what you're looking for is going to be a substrate. They're going to bind together. Or maybe your medium is going to be a antibody, and your product that you want to get is going to be an antigen, and they're going to stick together. So that's just to show you that we separate, but not based on the charge, based on sticking together. Or maybe you can have a sequence of a nucleotide sequence, and the nucleotide sequence you're looking for is going to be the opposite. So when you pour your solution, you will have the sticking of the medium to that through affinity to that particular substrate that you want to separate from your solution. There's also separation based on the polarity, and that is you can have a non-polar medium to dissolve non-polar substrate that you are looking for, or you can use a polar medium to dissolve a polar substrate. We will talk more about that. Okay, so this is now ion exchange chromatography. So the cation exchange chromatography will have a positively charged medium, and it will attract the negatively charged particles that we want to purify. And the anion exchange will be negatively charged, and it will attract for us positively charged amino acids that we want. So here the exchange is based on the charge, not based on the size, not based on the affinity. Now, this is size exclusion. That's very interesting. What's happening here, these are the beads. And the beads, like I told you, they have path through them. They have like avenues inside them. You don't want the red protein. You don't want the red protein. So the red proteins are so small, they're gonna be so busy going through the beads. You want the purple. The purple is too big to go through the beads, so it will only go through, be going between the beads. So that's why the large molecules will move between the beads and get out first. You can collect them first, while the red ones are going to be so trapped inside the beads or between the beads, so you cannot catch them early. It's gonna take them a long time to make it through, because now they're gonna get, these beads are blocking their passage and leading them to go through these zigzag paths. So these red ones, we don't want them. They're gonna come out later. It is the big ones who cannot fit through. They are the ones who zigzag and come out through the column, and we collect them in the eluate down here. This is affinity chromatography. And that is, like I said, when we use a medium, we use a medium that is like a nucleotide sequence or a nucleic acid, and that will bind the opposite complementary sequence. And that is now based, we have collected medium based on the affinity. And like I said, we don't only use DNA nucleotides, we can also use enzymes and substrates. We can also use antibodies, and we can also use antigens, as long as two things will bind together with affinity, like a lock and a key. Now, Western blotting, we mentioned in class, we use sodium diodysyl sulfate, and we put in the a chromium gel, we put all of the protein that we have purified and we run an electric current. This electric current is going to push our amino acids and our proteins farther from the site where all the electric current voltage is coming through, and all of the proteins are going to be running away from the voltage, and the heavier proteins will stay at the back, and the lighter proteins will make it all the way to the front. Then we will isolate those separated proteins, and sometimes we do antibodies to get them and isolate them, and then we can investigate them. So the detection uses labeled antibody. So in the end, we use antibodies, but there is no color change whatsoever. And this is the polyacrylamide gel that I talked about, that we use electric voltage. We run it through the gel, and the gel is going to allow for electric voltage to push the molecules, and then we separate them. So the voltage is pushing all the molecules, and all of the heavy ones will stay at the back, will stay in the back. So this is a protein sample. That's a protein sample. That's a protein sample. That's a protein sample. Protein sample. Protein sample. And when we run the voltage, the heavier ones will always be. So we put them and we watch their migration. We put them and we watch their migration, and we see that the heavier ones are always lagging in the back, while the lighter ones are always moving up to the front, because these are the largest, they stay at the back, and these are the smallest, they have moved. So we place all of our protein solutions in all these different wells. We run an electric voltage, and these guys get to stay at the back, and these are the smallest and the lightest ones. Then we remove them from the gel, and sometimes we use antibodies. Then we investigate them after we have already figured out their weight based on their separation. That's the gel, and these are the wells in which we put all of our proteins we want to separate. Okay, so this shows us the different properties of the agarose gel that allows for the movement to go from the largest molecules move the slowest, they stay lagging behind, and the lightest ones move to the front. Now we move on to hemoglobin. So hemoglobin is very important because the rate of oxygen diffusion is too slow for oxygen to arrive to my tissue. Tissue needs oxygen in large amounts due to its constant use of oxidative phosphorylation and the aerobic respiration. So the demand increases a lot, and fight or flight and worrying and emotional distress, that causes for an extra demand of oxygen. So you have to remember that the hemoglobin is very important because it is made up of 4 subunits. These 4 subunits are, 2 of them are alpha subunits and 2 are beta subunits. Each subunit of the 4, so the first alpha has 1 heme and 1 oxygen in the diatomic shape. Oxygen does not exist in O. So it has 4 of O2s. That doesn't mean it has 4 times 2, that is, it has 8 oxygens. No, the oxygen just comes like a double. That's just how it is. It's even when we use it for oxidative phosphorylation, it doesn't come monatomic, it comes diatomic. So it is 1 O2, not 1 O. So it has 2 alpha, 2 beta, 2 alpha, 2 beta. And each subunit has 1 heme group. So 2 alphas, each one has 1 heme. So 2 hemes. And 2 betas, each one has 1 heme. So 2 hemes, 2 additional hemes, a total of 4 subunits and 4 hemes. Each heme has 1 O2. So there are 4 hemes. That means there are 4 O2s. So that shows you 4 subunits, 1 heme on each subunit, and each heme has 1 O2 in the donut hole where the heme is at. You see, that's the donut hole. You see, the red ones are your alpha subunit, two red alpha subunit, two blue beta subunit. Each subunit has a heme, and each heme is binding an O2, 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 O2. Okay, the myoglobin, though, has only one subunit, does not have four. And this one subunit, just like the way the subunits for hemoglobin are structured, each subunit will have one heme and each heme will have one O2. So the myoglobin, so hemoglobin is like four myoglobins. So it has four subunits. Each subunit has one heme, meaning four hemes. Each heme has one diatomic oxygen for a total of four double atomic oxygens. So that's the structure of hemoglobin and myoglobin. That is myoglobin. See, myoglobin, one subunit, one heme, one O2 in the donut hole. Okay, there we go. So hemoglobin and the movement of oxygen. These are my alpha chains, and these are my beta chains. Each chain has this heme group, and the heme group has an iron, an Fe, a ferrous, Ferrous. Ferrous group is a Fe plus 2. Okay, it has two plus charges. While ferric is Ferric, it has Fe plus 3. So ferric that has more letters, ferric has 6 letters. And ferrous has Ferrous. It has 1, 2, 3, 4, 5, 6. It has 7 letters. So the ferrous with double, both of them have double R. So the ferrous with more letters has only 2 pluses. It has been oxidized twice. But ferric that has less letters, 3 letters, ferric, it is actually oxidized 3 times. So it has 3 pluses. And oxygen does not bind to the ferric iron with 3 pluses. It binds to ferrous with 2 pluses. So you can remember it this way. Oxygen is diatomic, O2, so it binds to Fe plus 2. That's the ferrous. Ferrous, more letters, less pluses, Fe plus 2, just like the number of the diatomic oxygen 2. So diatomic oxygen O2 binds to ferrous Fe plus 2. So that's the first ferrous, second ferrous, third ferrous, fourth ferrous, and it is on the heme. So the heme is this blue color, blue plaque, heme, blue plaque, heme, blue plaque, heme, blue plaque, heme, blue plaque. This iron center, the ferrous is the Fe plus 2, that's the yellow, Fe plus 2, yellow, Fe plus 2, yellow, Fe plus 2, yellow. That is where you're going to have your oxygen. And there is an additional protoporphyrin group. So heme has subunits. It has hemoglobin has globin is these are the globular units, the proteins, and it has a protoporphyrin. Don't forget the protoporphyrin ring. This is the ring that allows for the heme structure to happen. Don't forget the protoporphyrin. There's a question on the exam about what are the constituents and the structures of the hemoglobin, and these are the subunits, the protoporphyrin group, and the heme group with its iron center, the ferrous. There's a heme prosthetic group, the ferrous iron, methemoglobin won't work. Ferrous is when you have ferrous, that is the oxyhemoglobin. The methemoglobin is when you bind to ferric, that is Fe plus 3. Only Fe plus 2 bind oxygen, and that was on the exam, Fe plus 2. Okay, the hemoglobin and the movement of oxygen. So you know that in the structure of hemoglobin, we have one very helpful amino acid, and that is histidine. There's a question on the exam. What amino acid is found to confer stability to our hemoglobin structure, and that is histidine. Histidine movement is participating in the unloading of the oxygen. Yes, all we asked for was what is the name of the amino acid, and that was histidine. Okay, cooperativity, that's a form of positive feedback. So when hemoglobin enters the lung, it enters in the T state. T state meaning is empty, square, empty, does not have any oxygen. What happens now is one oxygen binds, that's O, circle. If one oxygen binds, it's going to facilitate and enable the addition of even more. So that we saw that also with the platelet plug formation. If you are injured, one platelet sticks, the second on top of it, the third on top of it, the fourth on top of it, the fifth on top of it. So that is known as cooperativity. So the binding of one oxygen to hemoglobin will lead to the binding of additional oxygens. The form of hemoglobin where you had zero oxygen, that's the T state. It is locked in a T state. A stable T state has no oxygen. And R state has all oxygen. This does not mean this does not mean you're gonna unload the oxygen or not. We're not talking about that. We're just describing. Is your hemoglobin full of oxygen? That's R. Is your hemoglobin not full of oxygen, empty, no oxygen? That's the T state. So no oxygen, T state. All oxygen, full of, that's the R state. So don't think of it as when you are in the tissue, you're gonna unload the oxygen, you're gonna be T. No, that's not true because there are factors like, for example, carbon monoxide. Carbon monoxide, when hemoglobin is so full of oxygen, it is so full of oxygen, it's right next to the tissue. You want to unload the oxygen to the tissue so the tissue can use it for oxidative phosphorylation and aerobic respiration. Carbon monoxide is going to stabilize the R state. It's going to lock it into this R state, telling, nope, hold on to your O oxygen. Do not let it go. And then you're going to suffocate to death. So that here, the oxygen bound and is affected by oxygen binding the R state. So here, the more oxygen bind, the less in the T state we're going to be, and the more we're going to shift close to the R state. So the more R you are, the more oxygen you have. The more T, the less oxygen you have. So the binding of the first oxygen favors the binding of the second oxygen. That is known as cooperativity. Cooperatively, is important as the hemoglobin rapidly passes through the lungs. So that's what we want in the lung. The lung is sending all the oxygen. So the hemoglobin is passing. So the lung says, here you go, that's one oxygen, that's two oxygen, that's three oxygen, that's four oxygen. So now as hemoglobin is rapidly passing through the lungs, it gets saturated. Okay. Hemoglobin and the movement of oxygen. So you see, entering the lungs, you are in the T state, no oxygen. As you move through, oxygen is more bound until it's all the way fully oxygenated. So the binding of the first oxygen favors the binding of the second. And the cooperativity is very important. This is the hemoglobin and the movement of oxygen. This here shows us the oxygen saturation, how much oxygen is saturated in my hemoglobin. So this here represents for me an R state. So I'm full of oxygen, 100% oxygen. Here I am, that's me. That's me right here. That's me right here, 100%. I am full of oxygen. That's R. I wish I can write. That's R here. That's R. Zero, I have no oxygen. That's T. That's T. So now look at hemoglobin. Hemoglobin, and so the y-axis represents how much oxygen is saturated in my molecule, whether hemoglobin or myoglobin. And the x-axis shows the oxygen saturation of the tissue. So here, if the tissue has 100% oxygen, then hemoglobin does not need to unload it. So that's why hemoglobin has 100% oxygen, because the tissue has 100% oxygen. Hemoglobin will say, listen, I am full of oxygen. I am 100% oxygen. And so are you. You're 100% oxygen too. So I don't need to give you nothing. So that's why it looks like that. But in the bottom, the tissue here will say, I'm suffocating. I'm suffocating. There is no oxygen for me. It's only 5%. So the hemoglobin will say, okay, I'm going to let go of 90%. And now it's going to let go of all the oxygen it has. And hemoglobin will only be like 10%, like
like 5% saturated with oxygen. And now it's going to become like a T state because it let go of 95. So you see, this represents the tissue. The more oxygen the tissue has, the more oxygen the hemoglobin has because it doesn't need to give it. The less oxygen the tissue has, the less oxygen hemoglobin will have because it's going to give it to the tissue. But look at myoglobin. Myoglobin doesn't care. Myoglobin, oxygen will say, the tissue will say, I'm 100% oxygen. Myoglobin will say, I don't need to give you, you already have 100% oxygen. Myoglobin will say, so yes, you have 100% oxygen. You don't need oxygen. But when we are here, the tissue will say, I'm 10% oxygen. Please give me 90% oxygen. Hemoglobin will say, sure, it will give it 90% oxygen. And now hemoglobin is broke. It has no oxygen. But look at myoglobin. At the same place, myoglobin is saturated with like 40%. It did give some, but it gave only 60%, and it still has 40% left. But look at myoglobin, it gave everything. Myoglobin, your dad, when you needed the money, he gave you everything. You needed the money, he gave you everything. But myoglobin, he's not going to give you anything. He's just going to give you 60%. So that's why myoglobin, if you examine the structure of myoglobin, he's like, is hyperbolic. And it shows that he has really strong affinity. You see, it's holding on to oxygen even when the tissue is dying. The tissue has 5%. He'll say, okay, I'll give you 60% and I'll hold on to 40. But hemoglobin will say you can take all of my oxygen you needed. So hemoglobin curve is sigmoid, like an S shape, while myoglobin is hyperbolic, indicating that the affinity for oxygen, the affinity of hemoglobin for oxygen and the saturation of hemoglobin for oxygen will be directly proportional to the tissue saturation of oxygen. If the tissue is really saturated with oxygen, so will hemoglobin be. It will have a lot of oxygen. But if the tissue is poor in oxygen, so will hemoglobin because it will give it all the oxygen it has. But myoglobin will say, no, I'm better for storage. I'm hyperbolic, better for storage. And hemoglobin is better for delivery. And the fetal hemoglobin is going to be right in between. It is a better giver of oxygen than myoglobin, but it will not be as efficient as hemoglobin. So there's a question on the exam asking you order them in levels of affinity for oxygen. Myoglobin has the most affinity because it's storage, followed by fetal hemoglobin because it does not bind to 2,3-BPG biphosphoglycerate, and it is not as efficient as hemoglobin, who will give all. So at low oxygen, you see myoglobin holds more than hemoglobin. You see, at low oxygen, hemoglobin has no oxygen. It let go all of that it has, but myoglobin has about 40% left. You are broke, you have no money, your dad gave you all the money he has, but myoglobin gave you only 60% and held on to the 40%. So it's like two banks. One bank, you have no money, will let you take all the money you need. And this one will say, nope, there is a limit. You can only take 60% and hold on to 40%. See? So at high oxygen, both hold 100% because the tissue doesn't need it. That's a sigmoidal binding curve. That's hyperbolic binding. Okay, so as the curve moves to the right, there will be less affinity for oxygen, meaning the situation is going to deteriorate. The metabolic demand is going to be worse. So when we shift to the right, we're going to have less affinity. When we shift to the left, we have more affinity. Remember that with myoglobin being at the leftmost because it has the highest affinity for oxygen, and hemoglobin being at the rightmost because it has the least affinity for oxygen. So the more the curve shifts to the right, the less affinity for oxygen you would have. The more we shift to the left, the less affinity for oxygen we're going to have. So myoglobin is better for storing oxygen, and hemoglobin is better for delivering oxygen. So that's the Bohr effect. The Bohr effect tells you as you shift to the right, hemoglobin is going to accommodate. So you see, originally, this is myoglobin. This is, I'm sorry, these are all hemoglobin, but that's showing you that the blue line is dealing with pH 7.4. The red line is showing you 7.2. That's more acidic. That's more painful. And this is 7.2 and carbon dioxide. So green is the worst. So this blue is just 7.4 acidic pH. Red is 7.2, even more acidic. Green is 7.2 acidic, and you have carbon dioxide, which is more toxic. So now that shows you that hemoglobin behavior in these three different mediums is going to be different. As the stress, so you agree with me that the stress increases from blue to red to green. Green is the most stressful. It is acidic with carbon dioxide. Red is less stressful because it's just acidic, 7.2. And blue is stressful, but it is 7.4. It's more alkaline than 7.2. So what's going to happen now is the graph is going to shift to the right so that hemoglobin can let go of oxygen. So at this, you see at the same, at this partial pressure, so when the tissue is only saturated with 20% oxygen, when the condition is blue, the hemoglobin is saturated with 0.3. That means it let go of zero point 0.7. That means it let go of zero point 0.7 here. That means it let go of zero point 0.7. It is holding on to 0.3. But when the situation is red, that means it's holding on to 0.2. That means let go of zero point 0.8. It let go of more oxygen. And now look at green when it is the worst. When it is the worst, you see that it's right here at 20. So here, this line, this dotted line represents the same partial pressure of oxygen, 20. So my tissue only has 20% oxygen. But in red, in blue, red, and green, not only do I just have 20% oxygen, I only have 20% oxygen and other problems. For blue, I only have 20% oxygen and 7.4 pH and no carbon dioxide. So hemoglobin will say, I'll give you zero point 0.7 and I'll hold on to 0.3. So right here in blue. In red, red will say, I'm also having 20% oxygen just like blue, but my situation is worse. I'm 7.2 pH and I'm more acidic. So hemoglobin will say, okay, yeah, your situation is worse, even though both of you are at the same oxygen saturation. So I'm gonna give red 0.8 and hold on to 0.2. So that's the fractional saturation of hemoglobin is 0.2. That means it gave off 0.8. On red, on blue, it gave 0.7. In orange, it gave 0.8. But look at green. Green will say, I'm worse than all of them. Even though all of us only have 20% oxygen, green will say, I have more acidity than both of them. I am very acidic and I have carbon dioxide. So hemoglobin will say, oh, you need a lot more oxygen. So I will give you now 8.5 oxygen and I will hold on to 0.15. So that shows you as you move right, as you move to the right, the affinity of hemoglobin for oxygen will be less, and hemoglobin will accommodate tissue with more stress by giving it more oxygen, and it will therefore, the hemoglobin will have less oxygen saturation because it gave away the oxygen that it was holding. So we will have a lot of T state. As the things get worse and worse, the hemoglobin will be more in the T state than in the R state. It will be less saturated with oxygen than before. So we shift to the right and we unload more oxygen than we used to accommodate for this excessive extra stress. Acids favor the release of oxygen from hemoglobin. Yes, because tissue that is metabolically deranged and that somebody who's running the marathon, there is a lot of lactic acid being secreted by the tissue. So all this lactic acid that is being secreted by the tissue, that is making the tissue environment very toxic. So when hemoglobin pass by, the acid is going to start pushing and displacing these hydrogen protons, these acids, hydrogen protons are going to start pushing and displacing oxygen away from hemoglobin. They're going to remove the R state of hemoglobin, R state full of oxygen, is going to make it T state. But remember, the hydrogen proton, it does not bind to hemoglobin. It just pushes the oxygen away from hemoglobin so the tissue can have that. Carbon dioxide, also the same way, like the acid, the carbon dioxide, because remember, carbon dioxide is carried in the blood in the form of carbonic acid. So carbon dioxide is a form of acid too. So that's why when you have acidosis, you'll start hyperventilating. So your respiratory system can throw away more carbon dioxide and decrease the acidity. So it favors the release of oxygen from hemoglobin. So that shows you that the acid and carbon dioxide are released by rapidly metabolizing tissue. So the tissue that is running the marathon, metabolizing so quickly, breaking down, catabolizing all of the glucose to give me ATP, it is building up so much metabolic waste like 2, 3 BPG, as well as carbon dioxide, as well as hydrogen proton, and these guys will just push oxygen away from the hemoglobin. And that shows you, you see, as we shift to the right, this the Bohr effect. You see pH, the pink is pH 7.5. Blue is 7.1. That's the most acidic. That's the most dangerous. So that's gonna cause less saturation of oxygen because hemoglobin is not going to be saturated. It's going to say, oh, blue needs oxygen. Let me unsaturate myself. Let me have low affinity for oxygen. Let me give off my oxygen. So it's going to have very low affinity in blue. It's going to have low affinity in green, low affinity in orange, and the least affinity for pink. So the more the situation deteriorates, the more I'm going to let go of oxygen. Less oxygen is bound at the same pressure. So that shows you less oxygen is bound at the same, you see, at the same pressure. When I change other stressful conditions, then my hemoglobin is going to change and accommodate that stressful condition and give you more. I gave you an example in class about what happened in the pandemic, and if everybody used to get paid the same salary, the people, if everybody gets used to get paid the same salary, now you have to, because there's extra stressful conditions, then you will be paid more. That's exactly what happened here. So as the pH went on, it dropped more and more and more. What happened now is that the oxygen, the hemoglobin will give more oxygen and become less saturated. So more oxygen required to have the same fraction bound. So you see that to the right, less affinity, to the left, more affinity. So the protons combine to the hemoglobin. The protons change the hemoglobin shape, and the reshaped hemoglobin loses the oxygen. So the acids do not bind here, that they favor the release. The acids favor the release of oxygen from hemoglobin. Carbon dioxide favor the release of oxygen from hemoglobin. But the hydrogen proton combine to hemoglobin, and the protons change the hemoglobin shape from the R state to the T state. So they're going to make it lose oxygen. And the reshaped hemoglobin loses the oxygen, and the rapidly metabolizing tissue would release these protons. So the tissue is suffocating, releasing protons, and hemoglobin is going to accept them and displace the oxygen in their place. So the rapidly metabolizing tissue get more oxygen from hemoglobin. So this is 2,3 BPG. That's the metabolic waste. That's the structure. You don't have to worry about its structure. You just need to understand that this is a toxic waste, toxic metabolic waste that is generated by rapidly metabolizing tissue. This is a byproduct of glycolysis, and exercising muscle cells rapidly use glycolysis, and exercising muscle cells produce acid, carbon dioxide, and these are the toxic things that promote for
of hemoglobin to release its bound oxygen and go from the R state full of oxygen into the T state with no oxygen. 2,3-BPG is a product of glycolysis. The exercising muscle cells rapidly use glycolysis. Fine. Okay, it locks hemoglobin in the T state. So the 2,3-BPG, when it sees that hemoglobin has oxygen in the 4 subunit in the R state, it will deplete it from all the oxygen, and it will have no oxygen, and that will lock it in the T state. 2,3-BPG and oxygen binding. Okay, hemoglobin and the movement of oxygen. So if the pH goes down, we're more acidic, 2,3-BPG increases, there's a lot of metabolic waste, and carbon dioxide increases, there's a lot of carbon dioxide, we shift to the right because we need to accommodate. That's really stressful. So the more you are to the right, the more stressful the tissue is, the more oxygen you need to give, and the less affinity hemoglobin needs to have for oxygen, and the more oxygen hemoglobin needs to give to the tissue. But if you move to the left, that means the pH is high, there is low 2,3-BPG, there is low carbon dioxide. That means the situation here is very nice, it's vacation, there's no need for no stress. So now the affinity for oxygen by hemoglobin will be more. So as you move to the left, there's not stressful conditions. We don't need to unload oxygen. So the graph is gonna shift to the left. But if there's a lot of stressful conditions, the graph is gonna shift to the right to depict that hemoglobin will be less saturated with oxygen because it's gonna give all the oxygen to the tissue. You cannot be saturated if you're giving away the oxygen. That's a slow metabolic rate. That represents slow metabolic rate because this represents fast metabolic rate, that you're running the marathon, you're getting so acidic lactic acid, so much metabolic waste being generated because you're burning so much carbohydrates and macromolecules to get ATP, and carbon dioxide is accumulated. So that's a very fast metabolic, stressful state. So rapidly metabolizing cells produce acid, and the rapidly metabolizing cells release carbon dioxide, and the rapidly metabolizing cells also release 2,3-BPG, and that's an example of a situation that favors the release of hemoglobin. So the rapidly metabolizing cells get more oxygen because they have these 3 things that favor hemoglobin to let go of its oxygen and go from an R state full of oxygen into a T state that has no oxygen. So 2,3-BPG is a big concern for smokers because the blood of smokers has high levels of 2,3-BPG. So the hemoglobin gets locked in the T state in passage through the lungs. So when the hemoglobin is coming to the lung and says to the lung, give me oxygen, fill me up with oxygen, I am in the T state, I'm all square, I have no O's, no oxygen, the 2,3-BPG will be so much, and you know that 2,3-BPG favors the T state. So in the tissue, when there's a lot of 2,3-BPG and hemoglobin arrives full of oxygens in the R state, the 2,3-BPG is going to displace all of the oxygens, and it's going to lock the hemoglobin in the T state, emptying it from the R state, T state. But the problem here, the same thing. When hemoglobin comes empty with no oxygen to the lung, 2,3-BPG will do the same, will say, no, just you cannot have oxygen. So the 2,3-BPG will impair, because that's what it does. It does not allow for hemoglobin to be saturated with oxygen. And that is meaningful in the tissue so that when oxygen cannot have a lot of oxygen and the tissue is suffocating. So 2,3-BPG will lock hemoglobin in the T state. It will change it from R state full of oxygen into T state no oxygen. But in the lung, the 2,3-BPG again is going to say to hemoglobin, oh, you cannot be in the R state. You have to be in the T state with no oxygen. So hemoglobin will say, I have no oxygen. I'm coming to load the oxygen.
but the 2,3-BPG will not allow it. And that's why the blood of the smokers, their blood is going to the lung, but because there is so much 2,3-BPG, it will lock the hemoglobin in the T state. It will not allow it to be saturated with oxygen and go in the R state. So the oxygen carrying capacity of the blood for smokers will be reduced. And carbon monoxide levels are also higher in smokers. So the carbon monoxide levels, they are very interesting because carbon monoxide will actually wait. It will wait until all the hemoglobin is actually full of oxygen and from the lung. So carbon monoxide will wait until hemoglobin goes to the lung and is converted from a T state, no oxygen, and becomes saturated with oxygen and becomes in the R state. But once the hemoglobin in the R state full of oxygen is arriving to the tissue, carbon monoxide will say, no, stay in the state of R, and it will not allow it to unload all the oxygen that it's bringing from the lung to the tissue. So carbon monoxide will lock and stabilize the hemoglobin in the R state. Okay, movement of carbon dioxide. So the rapidly metabolizing tissue is going to favor the displacement of oxygen. You see? Now, this was hemoglobin rich in oxygen. Okay, this hemoglobin that is rich in oxygen, when it arrives to a tissue that has a lot of hydrogen proton, it will let go of the oxygen to the tissue so the tissue can breathe, so the tissue can use the oxygen for oxidative phosphorylation and aerobic respiration. And instead, the hydrogen proton and the carbon dioxide will bind to hemoglobin. So hemoglobin came in as HBO, oxyhemoglobin, and then it let go of the oxygen, and now it's bound to HB, carbon dioxide, and the protons. This is very important for you to appreciate that hemoglobin that is rich in oxygen in the R state is going to, all hydrogen proton is going to remove the oxygen, and the carbon dioxide are going to remove, carbon dioxide are going to remove the oxygen, and they're going to bind instead. That's known as carbamino hemoglobin. This is oxyhemoglobin, hemoglobin with oxygen. That's carbamino hemoglobin when the carbon dioxide is bound to hemoglobin instead of the oxygen, because the tissue needs the oxygen to be given to the tissue and carbon dioxide to be removed from the tissue. Okay, so carbon monoxide and heme. So you see carbon monoxide, this is our oxyhemoglobin. It has oxygen, it has our protoporphyrin ring, it has our histidine group, it has our ferrous. And what happens is an additional histidine is present at the heme ion. It reduces the affinity to carbon monoxide, but it does not eliminate it. So carbon monoxide is found in a cigarette smoke. So note that the carbon dioxide does not bind to the heme, nor do the proteins. So that is in the state of carbon monoxide. So when there's carbon monoxide poisoning, usually the carbon dioxide and the protons bind to the hemoglobin and lead it to unload the oxygen and become less saturated with oxygen and has no affinity for oxygen and move from an R state full of oxygen to a T state with no oxygen in the tissue. But in carbon monoxide poisoning, carbon monoxide is not going to allow carbon dioxide to bind hemoglobin and let go of the oxygen, or bind the protons and let go of the oxygen. So it will lock. Carbon monoxide will prevent that, and it will lock it in the, it will lock it in the, uh, it will lock it in the R state. So fetal hemoglobin. Fetal hemoglobin, the body makes different globules over time, and the fetal hemoglobin is not alpha, two alphas and two betas, it's two alphas and two gammas. And these are the subunits that are found in the blood, but the one that is usually found, this is all not important. What's most important for you is to remember that fetal hemoglobin is
Adult hemoglobin is 2 alpha and 2 beta, while fetal hemoglobin is 2 alpha and 2 gammas. Fetal hemoglobin can bind to 2,3 biphosphoglycerate. It remains in the R state. You see, that shows you that fetal hemoglobin that is carrying oxygen, when it gets to the tissue, even though that there is 2,3-BPG that is supposed to displace oxygen, that will not happen. And that will cause fetal hemoglobin to remain in the R state, saturated, full of oxygen. Because if fetal hemoglobin did bind to 2,3-BPG, all the oxygen would be displaced out from the hemoglobin, and it will now become not in the R state full of oxygen, but in the T state. But that's not what happened to fetal hemoglobin, because it has 2 alpha and 2 gammas. It does not bind properly to 2,3-BPG, so it will stay locked in the R state. Okay, in sickle cell anemia. Sickle cell anemia is a genetic disease, and it's a uh, poly, single nucleotide polymorphism that leads to a mutation. The mutation is we needed the amino acid glutamic acid, GLU, but due to a mutation, we end up with valine. And that is causing the red blood cells to lose the rounded shape and form sickles. And this does not happen all the time. It's when you need them the most, as we mentioned in class. So patients who have sickle cell anemia at resting conditions, if you take a blood sample and you look at a blood smear, a blood spread, you can never tell that their red blood cells are different from somebody who has normal red blood cells. But the problem is, they exercise or when they are under some form of distress that requires more oxygen. Then the problem is that the problem becomes exacerbated, and then the cells start to sickle. So when you need them the most, as somebody very nicely in the class said, when you need oxygen the most, the patients who have sickle cell anemia, their red blood cells will start to sickle instead of looking discoid and biconcave. So these are sickle cells. They look like a sickle like that. So the change is caused by the polymerization of hemoglobin, and that happens when there is low oxygen levels. So the rounded cells move easily through the capillaries. So that's for a normal red blood cell. It moves through. But the problem is the sickle cells, they start to sickle and aggregate, and they clog the blood vessels. So the sickle cells are removed by the spleen and destroyed by the spleen. That's why the spleen is the graveyard of the blood vessels. Patients who have sickle cell anemia, they actually have a benefit, but not the homozygous, recessive or dominant. It is only the heterozygous. So the benefit of sickle cell mutation is for the heterozygous, not for the homozygous recessive or the dominant. Hemoglobin and the movement of oxygen. So animals have widely varying oxygen needs, ATP generated much more efficiently in the presence of oxygen due to oxidative phosphorylation and aerobic respiration. Hemoglobin and myoglobin are related, but they have different functions. Hemoglobin has 4 subunits and hemes, and myoglobin has one of each. Myoglobin has one of each. We talked about that. Binding of oxygen by hemes, iron pulls up on the histidine, and the changing of hemoglobin shape converts hemoglobin from T state to R state to accept more oxygen. The R state binds oxygen better, and the T state releases oxygen better. So R state, when we are fully bound with oxygen, all O's. T state, when we have released oxygen, we are all square. In the Bohr effect, the binding of carbon dioxide and hydrogen protons favors the oxygen release. So these stressful conditions promote for more release of oxygen. The Bohr effect explains how the oxygen and carbon dioxide exchange in the lungs. 2,3-BPG is a metabolic waste that is produced by rapidly metabolizing cells. It too favors oxygen release, and the fetal hemoglobin can bind 2,3-BPG and has greater affinity than adult hemoglobin. Sickle cell anemia is a genetic disease of hemoglobin. Okay. In low oxygen concentration, red blood cells of sickle cell anemia suffers from sickle cell. They form sickle shapes. The sickle cells stick in capillaries and can be fatal. And people heterozygous for the mutated gene survive malaria better than others. And that's the protein structure. We talked about the primary sequence of the amino acid. The secondary sequence is the interaction of the amino acid that is close in the primary sequence. The tertiary structure. So this is the primary structure like beads, the alpha helix and the beta sheet. These are your secondary structures. And the alpha helix is made by hydrogen bonding, and the beta sheets also by extensive hydrogen bonding. Then we move from secondary into tertiary by disulfide bridges, extensive hydrogen bonding, hydrophobic effects, van der Waals interactions by disulfide bridges, and then the quaternary structure. These are complex of protein molecules. These are the most complex, and they are made up of attached subunits with ionic, in addition, ionic bonding. So quaternary, there's interaction of quaternary is represented by interaction of the protein subunits. So that is the substrate active site. This is the enzyme that is binding the active site. This is the active site on which the substrate would bind. So the active site is the site of the enzyme where the substrate would bind. So this black structure here is the substrate, and the blue site is the active site of the enzyme. Okay, it's very interesting to know that if we want to go from substrate to product, there is an activation energy barrier that we have to go up like a hill, like a mountain. So the enzyme will catalyze the reaction by decreasing the activation energy barrier. It does not affect the total free energy. It does not affect the overall free energy. You see, it's the same, whether you, how much, it doesn't matter how much you drop the activation energy barrier. So the enzyme decreases the activation energy necessary for the reaction to go from reactants to products. But the net free energy remains the same. Also, the reactants do not get consumed in the reaction, and the enzyme does not get consumed in the reaction. The enzyme does not give you different products, and the enzyme catalyzes the reaction in both directions, in the forward and the reverse direction. See, that's the reaction that goes forward and the reaction that goes backward. And it does that because it has the path, the passage to the transition state complex that is here. This is the saddle point. That's the top point. This is the saddle point. Okay, enzymatic reactions. Enzymatic reactions lower the activation energy. Enzymes catalyze reversible reactions in both directions. Enzymes do not change the overall energy. Enzymes do not change the equilibrium concentration. Enzymes speed achieving the equilibrium. So in the types of the reactions, you have a single substrate that will give you a single product, single substrate that can give you multiple different products. You can have multiple substrate that will give you single product. And sometimes you can have multiple substrate that will give you multiple products. So the enzyme, if you use it here, you're supposed to get, you have one substrate that will give you two products. It's not gonna give you here. One substrate will give you one product. No, it will do the exact reaction. Just make it faster. It will not affect or change the reaction. Okay, enzymes. Here, you see that this is the enzyme. And the more substrates you add, the more enzymes are going to be able to work. So substrates actually help out. The more substrate you add, the more the reaction rate is going to increase. So imagine you have like a 10 shuttle buses outside the subway station, and nobody is getting out the subway. These shuttle buses are stopped. They cannot move. There's 7 of them. So when 100 people come out, the more people who take the bus, the more the buses are going to start up and they're going to start moving. So you will see an action that's happening. So in the beginning, if 100 people come out, the buses are going to get full. But if you have like 5,000 people coming out,
then all the buses are gonna already be filled. You're gonna reach, you're gonna level off. You're gonna reach a level where it doesn't matter how many people come out for the shuttle buses, they're already full. So same thing. If here the shuttle buses are empty, so the more substrates you have, the more activity you're gonna have. But you're gonna reach a point where the more you add substrates, now all the active sites have been filled. So now you're gonna level off at a level known as the ceiling effect, and it doesn't matter how much more enzyme uh, substrate you add, the reaction rate is not going to change. That's what we see here. So that's the reaction rate, how fast the reaction is going. That's the substrate concentration. So in the beginning, the more you add substrate concentration, the more you're going to have the reaction go faster until you reach a point like here. Now, it doesn't matter how much substrate you add, the speed is not going to go up, it's going to level up because all the shuttle buses are full. The Km is the substrate concentration that is going to correspond to half the maximal speed at which the reaction is going to go. And we said that this Km is has an inverse relationship with the affinity of the enzyme to the substrate. So the higher the Km, the lower the affinity. And the lower the Km, the higher the affinity of the substrate to the enzyme. Okay, we have it. All of this is this is the maximal speed occurs when an enzyme is saturated by the substrate. That's called the Vmax. And that is also dependent on the amount of the substrate that is added. And the Km is a measure of the enzyme's affinity for its substrate. It is inversely related to affinity, as we mentioned. So the higher the Km, the lower the affinity. The lower the Km, the higher the affinity. There we go. The higher the Km, the lower the affinity. The lower the Km, the higher the affinity. That's the normal substrate. And that's, okay, competitive inhibitor binds to the same site, the active site that the substrate binds to. So this is the substrate. You see how it looks like? The competitive inhibitor looks just like it. So it competes with the substrate to the same binding site. So how can we, if there's a competitor, how can we defeat the competitor? So the competitive inhibitor resembles the natural substrate and it competes with it for the binding site of the active site. So if you add more substrate, more yellow, you're gonna have more chance of the substrate binding to the active site. Okay, so let's say you're playing a game where there is about 50 seats, and these 50 seats can either be occupied by 50, they can only be occupied by 50 individuals, and you have 50 Marine soldiers and 50 Navy soldiers. And there's 50 seats. You have 50 Navy, 50 Marines. And you tell them, go and sit. Obviously, it's going to be closed. There's going to be 25 Navy, 25 um, Army, for example, or maybe 26 to 24, for example, something like that. But let's say now you change the competition where you only have 25 Army soldiers where you have now, for example, I'm sorry, you have 50 seats, and now you have 50 Army soldiers, but you don't have 50 Navy, you have 100 Navy. Who do you think will now occupy more seats? Because now you have more of the substrate, you're gonna defeat the competitor. So if we have more yellow substrate, we're going to have less green competitive inhibitor. So that's the competitive inhibitor. There is another kind of inhibitor known as the non-competitive inhibitor. The non-competitive inhibitor does not bind to the same active site. It binds to another active. It binds to another site, known as the allosteric site. So for non-competitive inhibitor, it does not matter how much substrate you add because the substrate does not bind to the allosteric site. The substrate only binds to the active site. So for competitive inhibitor, because it binds the same active site as the normal substrate,
The more substrate you add, the more you're going to defeat the competitive inhibitor, and the less the competitive inhibitor is going to bind to the substrate active site, and the more the normal substrate is going to overcome the competitive inhibitor. But if you are competing with a non-competitive inhibitor, then it will bind to another site. Then it doesn't matter how much more substrate you add, you will never be able to overcome the non-competitive inhibitor because it's not competing with you to the active site. It's already binding to an allosteric site. Okay. You see, that's the non-competitive inhibitor. It does not bind to the active site. It binds to the back, so to an allosteric site. So it does not matter how much more you. So the point you need to know is here for the competitive inhibitor, the more substrate you add, the more you're going to defeat the competitive inhibitor and you're going to bind the substrate active site. But for the non-competitive inhibitor, it doesn't matter how much more substrate you add, you will never be able to overcome a non-competitive inhibitor because it's not competing with you to the active site. It's binding to the allosteric site. So non-competitive inhibitors do not resemble the substrate and do not compete with it for the active site. Instead, they affect the enzymes by binding a different location on the enzyme. There are different classes of enzymes. There are the oxidoreductases. They catalyze. So the enzymes are all biocatalysts. They speed up reactions. Some of them speed up oxidation reduction reaction known as oxidoreductases. Always they end with aces. Transferases, they transfer a functional group like a methyl group or a phosphate group. So when they are activated, these transferase enzymes, they transfer a functional group or like a methyl or a phosphate group. Hydrolases, they hydrolyze the bond, they break the bond. Lysases, they are non-hydrolytic and they are non-oxidative, but they break the bonds. So these are just examples of different enzymes that you don't have to know them, but just remember that isomerases, they isomerize, they rearrange the structure. Ligases join two molecules by making covalent bonds. So that shows you what happens with oxidoreductases. They change they perform oxidation. So this one has a positive charge. It gained an electron. It became neutral. Transferases, like a hexokinase, it added a phosphate group. You see, it added a phosphate group. So it transfer a functional group like a phosphate group. Hydrolases break the bond with water. You see, like a peptide bond, you add an OH here and an H on the other side. And the lyases, they break the bond, but without water. They just cleave the bond without water. And then there are the isomerases, like you see, phosphoglucoisomerase. It converted for us glucose 6-phosphate. It has the same number of carbons, the same number of hydrogens. They have the same structure. It just rearranged it. So isomerases catalyze the isomeration changes or the rearrangement. So glucose 6-phosphate is a different rearrangement of fructose 6-phosphate. So if you count the number of carbons, they are the same in both molecules. They are just rearranged differently. That's known as isomerization. Ligases. Ligases join two molecules by making a covalent bond, like in here. You see, the ligase, it connected L-citrulline to a phosphate group from ATP. It connected the L-citrulline to ATP. Then it connected aspartate to the L-citrulline. You see, now you have the pink aspartate connected to the L-citrulline this was mediated by the ATP. So that just shows you that ligases join two molecules by making covalent bond. We've seen this ligases in the DNA replication when we join together the Okazaki fragments. Okay. Central dogma of biology is we replicate the DNA using a DNA-dependent DNA polymerase, and then we transcribe the DNA through an RNA, through a DNA-dependent RNA polymerase. Then we translate the proteins, the structural and functional proteins of interest. So now let's look at, the exam was centered around these points. Let's look at the exam. Been talking for how long now? Okay. All right. So let's go through all the questions very quickly. Okay. So from the answer choices below, this question is asking you, which amino acids have hydroxyl groups? Which amino acids have hydroxyl group? You can see that the amino acids that have hydroxyl groups are your, so hydroxyl is an OH. The amino acid that has a hydroxyl group, like serine. Serine has an alanine does not have an OH. Does alanine have an OH? No. Tryptophan does not have what is tryptophan? I'm sure tryptophan does not have an OH. So we just look at the R group. The question is, does that, when it says from the answers choices below, the amino acids with hydroxyl group are, so meaning which amino acids that have R groups that have hydroxyl groups in them. So these are the ones, tyrosine, and serine, tyrosine, and there's no tyrosine. There is here there is no they only have serine and threonine. So here serine, threonine, tyrosine. Okay, these are the three amino acids that have OH. From them, they only have two, serine and threonine. Question number two, from the amino acids below, the acidic amino acids. We said that the acidic amino acids are the ones that have a OH. They are the ones who are able, to donate a hydrogen, to donate a hydrogen group for us. And that will be these guys, these acidic groups. They are able to donate a hydrogen. So that is aspartic acid, and aspartic acid is known as aspartate. So this aspartame is confusing because there's a substance known as aspartame, but it's not aspartate, it's aspartate. So I will give credit to everybody who just clicked aspartame thinking that it's aspartic acid. And glutamate is another way of saying glutamic acid, and that's our um answer. These two are very important acidic amino acid that donate hydrogen when we need them. Assuming all other conditions are equal, which of the following amino acids is expected to have the most neutral charge? Most neutral charge at physiological pH, that's a Zwitter ion. A Zwitter ion means that at physiological pH, they will be dipolar, but they will cancel each other. Like perfect example is this non-polar amino acid. That's a minus. That's a plus. They cancel each other. The glycine has no charge. That's a perfect Zwitter ion, and they are neutral. So this question is asking you which of the following amino acid is expected, assuming all other conditions are equal, which of the following amino acids is expected to have the most neutral charge at physiologic pH? Which is another way of saying which of the following amino acids is expected to be a Zwitter ion. A Zwitter ion meaning you're dipolar, but your two charges cancel out. And the R group has no additional charge. So this here, the positively charged, can never be a Zwitter ion, because when these two cancel out, when this minus group in the carboxylic terminal and the plus group in the amino terminal cancel out, minus and plus cancel out, there is an overall plus group here with the nitrogen. That's a plus charge. That's why it's positively charged. Here, this is minus negatively charged because these two cancel out, and this group will incur an overall negative charge. Okay, so these guys here are neutral. These guys here are neutral. They're also Zwitter ions. They're also Zwitter ions because, yes, hi. Yeah, I'm closing in about an hour. Yes, I have a Alexa now. Kadir, Rod Kadir. Professor Kadir. Yes. All right. Thank you. So the Zwitter ion are two different. They are two different. They are dipolar. These are amino acids that are dipolar, but they cancel out, and the remaining R group will be neutral. So this is an example of Zwitter ion. This is an example of Zwitter ion. Aspartate, no. that is going to be negatively charged. That's not going to be neutral. Lysine, if you look at lysine, it has a positively charged. And if you look at histidine, histidine will have an overall positive charge. But when you look at ASN, that is known as asparagine. That's a perfect example of a zwitterion who is overall neutrally charged when these two guys cancel out, with the carboxylic terminal and the amino terminal cancel out. It will have no overall charge. What are the four classes of amino acids? So it should be polar neutral, like we said in the group, polar neutral, non-polar, polar positively charged, or polar negatively charged. So uh, polar neutral, non-polar, as it should be non-polar, polar, acidic, and basic. This one, that's perfect. Non-polar, polar, and polar acidic, and polar basic. So that's the right answer. A peptide bond is formed between a carboxylic group and amino group. We have seen that. Polypeptides are molecules that contain multiple amino acids. So we've seen that they contain multiple amino acids. Which of the following amino acids contain a hydrophobic functional group? So that they're asking you which one is not, so that if you have a hydrophobic group, you're all hydrocarbons, hydrogen and carbon, hydrogen and carbon. So that's the R group. So they're asking you which of the following are non-polar. Serine is polar, so that's not hydrophobic, that's hydrophilic, and that's lipophobic. But these guys, the hydrophobic, they are lipophilic and hydrophobic. So valine, perfect, that's nonpolar. That will not dissolve in water, that will dissolve in lipid. So the valine, the phenylalanine, these are perfect examples. Carbon monoxide binds to hemoglobin in our red blood cells and displaces oxygen. This contributes to shortness of breath in people who smoke. That's perfect. That's an example of how carbon monoxide locks hemoglobin in the R state, not allowing it to become T state by removing all the oxygen. So if you go to that's question number, let's go quickly. Who are the four? Make sure that the answers have been placed in. That is true. Okay. In sickle cell anemia, the crescent-shaped red blood cells are as a result of a substitution of a single amino acid. We said single nucleotide polymorphism, and that caused the mutation, and we were supposed to have glutamic acid, and we ended up with valine. So the answer is glu to val, glutamic acid to valine. Okay, how many heme groups is or are in hemoglobin molecule? So if you look at the answer choices, how many heme groups? So we said there are 4 subunits. Each subunit has 1 heme, and each heme has 1 diatomic O2. So how many heme groups? There is heme in each subunit. There is 4 subunits, 2 alphas and 2 betas. That means there are 4 hemes. Altogether, hemoglobin combined a total of how many oxygen? So it combined, it has 4 subunits. Each subunit has 1 heme. Each heme binds 1 O2. There is 4 O2s. There is 4 O2s total because each subunit has 1 O2. There is 4 subunits. Each subunit has 1 heme. Each heme binds 1 O2. That's a total of 4 subunits, 4 hemes, and 4 O2s. All of the amino acids in the acid choices below are zwitterions, except these guys on the positively charged and the negatively charged. Let's see what we have. Let's see what we have. So proline, valine, lysine, these guys are fine, or these guys are fine, except arginine or anybody in green or purple. What characteristics is shared by hemoglobin and myoglobin? Both of them are but they are both globular. What question number is that? They are both. What characteristics are shared by myoglobin and hemoglobin? They both have closely related primary. They both are globular proteins containing a common amino acid, porphyrin. Excellent. They both have porphyrin and iron. They both have porphyrin and iron. Question 14. The phenomenon that allows the hemoglobin to bind to oxygen where it is in abundance and release it where it is less known, that is known as cooperativity. We talked about that. When myoglobin is denatured using heat, denatured meaning that we break apart every weak bond, ionic bond, hydrophobic interactions, van der Waals bonds, hydrogen bonds, except the peptide bond. Its amino acid composition will change? Nope. It will be intact. The C terminal will change. Nope. The amino acid sequence will change. Nope. The tertiary structure will change. Yep. That's the answer. When the amino, when hemoglobin is treated with hydrochloric acid, its molecular weight will be unchanged. Quaternary structure will be unchanged. The conformation will be unchanged. Primary structure will be unchanged. Yes, because this hydrochloric acid will destroy all of these. Denaturing will only happen from quaternary to tertiary to secondary, destroying all these hydrogen bonds, all of these weak hydrogen bonds, these weak hydrophobic interactions, these weak ionic bonds, and so on. And disulfide bridges, but definitely not the primary sequence. Okay, gel filtration, chromatography of proteins. Which of the following is true? Okay, so this may require us to go here. This is ion exchange chromatography. In ion exchange chromatography, what happens is we have a resin of a particular charge, and we put through our protein of interest. So we want green, and we put an affinity tag, this orange tag, and our resin have an affinity, will have an ion change. And what happens is when you put all of your protein of interest, the resin will be positively charged, and your protein of interest will be negatively charged, and it will be based on charge. So ion exchange chromatography is based on charge, based on based on charge affinity. It's based on charge bonding. It bonds with the charges. But when we look at affinity chromatography, okay, this is mixed up. This one is affinity chromatography. This one is affinity chromatography. And the one below is ion exchange. The one below is ion exchange chromatography. This one is ion exchange chromatography, because you see here, this is a cation exchange, that's an anion. Cation meaning I need to catch a cation. You see, what I need is the desired charge. These are my desired charges, plus and minus. The undesired, these are the opposite. And that is my buffer, and that's my charged resin. So if I want something positively charged, my medium would all be negative. So it can bind to it and catch it from me. Then I wash with my buffer, and all the stuff that I don't need, if it's minus charge, then the minus, if the cation exchange means I need to catch cation plus charge. So the medium will be negative, so negative can bind the positive. But the negatively charged proteins will be repelled and they will exit out. We don't need them. You see, they will be repelled. But if I need something that is negatively charged, I use a positively charged medium, and it will bind that negatively charged protein, and everything will be washed out, and the remaining will be washed out. This is ion exchange chromatography. And this is affinity, where I have an affinity that is based on some enzyme. Some enzyme will bind the substrate, or antibody will bind an antigen, or a nucleic acid will have another nucleic acid sequence to catch it. So I have some affinity that is based on antibody antigen interactions, or enzyme substrate interactions, or maybe nucleic acid sequence that is dependent on some form of nucleic acid sequence. This is gel filtration. So in gel, you have beads, and these beads have holes through them. They have holes through them. What I need will be going through the sides, will be going through the sides of the, maybe I have the picture, the photo somewhere here. The photo, I can have the photo somewhere. Okay. Well, you see these little zigzag lines. They will allow for small particles to go through, and that will waste time while the big particles will go through the gaps. And then in the end, I will have my solution that I want to get out. So this is based on size. If you're small, you will be trapped in the middle. But if you're large, you will zigzag your way through and you will exit, and I will collect you in the bottom. Salting in and salting out, what happens is you add salt to some protein that you want to collect. The more salt you add, the more the protein is going to get cornered, like here, until when you add so much salt, your protein is going to crystallize and come out solution because they are so sequestered, they are so tightly locked in to each other, they're going to start clumping out. That's called salting out. So now let's go back to our question. Our question says the gel filtration chromatography. Which of the following is true? Large or elongated proteins enter the pores. No, it is the small ones who enter the pores. Small proteins enter the pores in the beads. That's true. That is true. The small proteins enter the pores in the beads. Small proteins elute from the bottom of the column first. No, they elute in the end because they are so busy going through the zigzag lines. But in the meantime, the big proteins have already escaped through the gaps between the beads. Large elongated proteins elute from the bottom of the column later, and that is not true. They elute first. So let's go to the next question, which um match. Now let's do the matching. In the matching, 2,3-BPG stabilizes the state of hemoglobin. Yeah, T, it will change oxygen R from R into T. It will remove all the oxygen so that because it's secreted in the tissue, the 2,3-BPG is a toxic metabolic waste of the tissue that will displace all the oxygen from hemoglobin, who is in the R state full of oxygen, and displace it to the tissue. Now it will become T state. Hemoglobin oxygen curve is hyperbolic. Oxygen binding results in this state of hemoglobin. Yes, it's going to be R, and hemoglobin oxygen binding curve has this shape known as sigmoidal. Okay, the fetal hemoglobin has an affinity for 2,3-BPG. We said that it does not. It does not have an affinity for 2,3-BPG. It does not have an affinity for 2,3-BPG. That's why it holds on more to the oxygen. It's better for storage than adult hemoglobin. Carbon monoxide stabilizes the R state. It does not allow hemoglobin to bind to the T state. Match the following protein purification techniques with the basis. So now let's look. Affinity chromatography, like I said, we bind through like binding affinity of some nucleic acid to the complementary nucleic acid sequence, or some enzyme who's going to bind to another substrate, or some antigen is going to bind to some amino acid. Ion exchange is based on the charge. If you are positively charged, you will attract negatively charged proteins of interest, and all the other proteins are going to go out. Gel filtration is based on size. The small protein will zigzag through the beads, while the large proteins will come out quickly by going through between the beads. Salting in and salting out is based on the solubility. The more salt you add, the more your protein of interest is going to crystallize. So each protein has like a fingerprint, has a particular salt that, for example, fibrin, the more salt you add if you add 0.8 molars of potassium sulfate, then fibrinogen is going to get salted out and crystalled out. So all you need to know is that it's based on solubility. Which of the following protein detection methods produces color change? We talked about that. That's ELISA. So in ELISA, what happens is we saturate our medium with antibodies. Then we drop our antigens of interest. Then we watch this interaction between the antibody binding the antigen. Then we add some enzyme linked antibodies that when they bind the antigen, they will, when they bind the substrate, what happens is there will be a color change in the solution. So that is the mechanism that requires a color change. Which of the following protein detection methods requires sodium dodecyl sulfate page? And that was the Western blot. Western blot, we said we put in the well all of the proteins of interest and we project electric voltage that causes all the proteins to propel and move forward. And the heavier proteins will stay in the back, lighter proteins will move to the front. The unvarying, varying meaning it will not change. Varying meaning it will change. Unvarying, it will not change. So that means no matter what cell you will have the same DNA. If you are a skeletal muscle, a neuron, a liver cell, hepatocyte, if you are a kidney cell, if you are a bone cell, if you are a cell of the muscle of the heart, if you are a blood vessel cell, you will have different proteins. This is definitely different. This is varying. The proteins inside the heart cell, the proteins inside the muscle cell, the protein inside the nerve cell, the protein inside the pancreatic cell, they're all different, but the DNA is the same. So we do the central dogma of biology, and there are different polymerases that will transcribe different regions of the genome to give us the different proteins that will make the cells achieve their structure and function. Protein is a polymer of amino acids. In the amino acid structure, the differs from one amino acid to another. That's the R group. The R group is going to be different. That R group is what makes one amino acid acidic, the other amino acid basic. The structure below has how many peptide bonds? You see, that's one carboxylic group, that's the carbonyl, and that's the amino group. That's one, and that's two of them. Okay. Uh, the structure below contains glycine. Glycine looks like that. So you need to find an alpha carbon that has two H's. So where are my alpha carbons? If these are my peptide bond, then that's my alpha carbon. No, it does not have two H's. This is my alpha carbon. No, it does not have two H's. And this is my third alpha carbon. It does not have two H's. No, it does not have glycine. Which of the following statement is correct regarding the structure below? Okay, so what are the statements? Let's see. Okay, there is one acidic group. In order to be acidic, you have to be one of these guys. Okay, aspartic acid and glutamic acid. Does it have aspartic acid and glutamic acid? No, the R groups, what are my R groups? These are, this is my amino terminus, that's my H, that's my R group. This R group is not an acid. This R group here is not an acid. And this R group here is not an acid. So nope, it does not have an acidic group. There's methionine. So let's go look at what methionine looks like. This is methionine, two carbons, S, and a third carbon. You see, two carbons, two carbons, S, and a third carbon. Yep, that's perfectly true. There is no acidic groups. Are there no acidic groups? There are no acidic groups. Nope, we saw that there were no acidic groups, so that's true. The structure can be broken down by hydrolysis. Yes, if we add OH to this here and H here, we break this peptide bond. We know that we break peptide bonds by hydrolysis. We know that. We talked about that. So that's perfectly true. The structure can be broken down using heat. Nope. This is a primary structure. It cannot be broken down using heat. If it was a tertiary, secondary structure, it could be broken down using heat. There is no hydrophobic groups. Is that true? There is no hydrophobic group. Is that true? There is no hydrophobic group. No, that is not true because for the time being, this sulfur group is bind this methionine. You see this methionine, it is already bounded to two carbon. It cannot have a third bond. It is definitely a non-polar group. But that's not true. There's a hydrophobic group. That's not true. And the other guys, they are this one. It cannot it cannot accept any more hydrogen, so it's also hydrophobic. And these guys, they can accept. So this is here is a polar group. So there are polar and there are non-polar groups. The structure was formed through dehydration. Yes, a peptide bond is made by dehydration by removing an OH from carboxylic and H from the amino terminal. The structure cannot be destroyed by denaturation. Exactly. This structure cannot be broken down except by hydrolysis. Denaturation is only good enough to break down hydrogen bonds and ionic bonds and hydrophobic effects, salt bridges, and disulfide bonds. And that is found in going from tertiary to secondary or quaternary, all of that stuff. There is cysteine. There is cysteine. So this is cysteine. So cysteine is not true because we don't have cysteine. Cysteine, you must have the thiol group attached to carbon and a hydrogen. But here it is attached to carbon and another carbon, which makes it methionine. All the amino acids in the acid choices have chiral alpha carbon, except we know that there is nobody besides glycine that is achiral. All of them are different, except glycine has two hydrogens. Three-letter abbreviation of tyrosine doesn't get easier than that. Tyrosine has TYR. Okay, the polypeptide, lysine, glycine. So the leftmost, that's the amino terminus. The rightmost, that's the carboxylic terminal. So the amino terminal is going to be the lysine. Methionine is more likely to form disulfide bridges because it contains sulfur. That is not true. It is the cysteine, not the methionine. You see, methionine is already preoccupied with two bonds. It already has two bonds. It cannot form disulfide bridges because it has a bond with a carbon and another carbon. It's in the middle of a carbon and another carbon. But the cysteine is in the middle of a carbon and another hydrogen. That hydrogen is easy to let go of. So it is the cysteine will remove this hydrogen and bind to another sulfur of another cysteine. Okay. Which of the following can be used to describe the C terminus? So the C terminus is the carboxylic terminal. What are the choices? Let's just see the choices. It is acidic, yes, because it gets deprotonated, it gets a negative charge, it gives its hydrogen proton when we need it. If the situation are too acidic, we talked about that. It is to the right, and it's always to the end because the beginning is the amino terminal. Okay, it is not basic. It is not found to the left as the amino, and it is the amino terminal that is found to the left, and that's at the beginning of the polypeptide chain. So this question says it requires critical thinking. Proteins can have a maximum of 4 levels of structure: primary, secondary, tertiary, quaternary. Although the proteins can spontaneously fold to a functional conformation, there are a variety of denaturing agents that can be used to disrupt the folding strategies of proteins. Mercaptoethanol is an example of protein denaturing agent. We said that's the one that breaks disulfide bridges. Its mechanism for dismantling proteins to disrupt the disulfide bonds found in the proteins. When urea is introduced, urea is like mercaptoethanol, but it does not break disulfide bridges. It breaks the hydrogen bonds holding the proteins together. So if you apply urea to an alpha helix, you will go right back to a primary structure because it breaks all the hydrogen bonds. So the heat can also be considered denaturing agent, which has the potential to disrupt all molecular interactions in a protein. Based on the information, which of the following proteins would be least affected by the introduction of mercaptoethanol? So if a protein has no cysteine, it cannot have disulfide bridges, and if it does not have disulfide bridges, mercaptoethanol will only break disulfide bridge. So the mercaptoethanol will not affect proteins that have no disulfide bridges. A protein that has no cysteine amino acid residues, yep. Which of the following most accurately describe the central dogma of biology, going from DNA to RNA to an amino acid? We live in an ever changing environment outside of our body. Our internal yet our internal conditions remain relatively stable. This is called homeostasis. Homeo means the same. Stasis means constant, which means staying constant by staying the same. So no matter if temperature increase outside, we secrete sweat to cool down. And if temperatures if temperature is so low outside, it's so cold, we shiver to warm ourselves up. So that's homeostasis. Metastasis is when cancer moves following state of invasion. The cancer cells invade the baseline membrane of the tissue that they originated from, and they travel through the blood from like the bone, they travel all the way to the lungs. Responsiveness is a part of the adaptive mechanisms that lead to homeostasis, but this is not the collective when we have stable internal condition. That is the feature of homeostasis. Adaptation and all of that. These are the steps that we need to achieve in order to maintain homeostasis. Hemostasis and hemostasis are tomato of the same thing, which is hemostasis, that is to stop the loss of blood. So hemo is blood, and stasis is having a constant blood supply, and that is the coagulation of the blood. You don't want to lose the blood. Where does oxidative phosphorylation aerobic respiration take place? That's in the mitochondria through the electron transport chain, and we will learn more about that. Cells that are damaged beyond salvage must, for the health and well-being of the tissue, must initiate cell suicide or programmed cell death through the actions of the mitochondria. This is a process that is called what? This is a process that is called apoptosis. Apoptosis. Necrosis is when the cell dies, but not with apoptotic cell death. When it dies due to some damage and there is no oxygen, and the mitochondria is the one who is responsible for initiating apoptosis. So the mitochondria didn't have time because the mitochondria needs oxygen to perform oxidative phosphorylation and then initiate apoptosis. But it didn't have time to do that, so the cell died prematurely and the membrane integrity was dismantled, and all of the lysosomal enzymes spill out to destroy the nearby tissue. That what leads to the uh, inflammation. Autophagy is when cell eats itself alive. These are all different topics that have nothing to do with cell death, and that is programmed cell death, programmed by the mitochondria. Mitochondria program cell death. That is apoptosis. Exercise, emotional distress, and fight or flight lead to, what do they lead to? They lead to increased blood carbon dioxide. Let's go here. Exercise. Exercise, emotional distress, shock, fight or flight, lead to increased carbon dioxide, decreased oxygen, high hydrogen proton, low pH, high acidity, and high OH. No, the OH will be decreased. There will be low OH, and there will be low 2,3 BPG. Nope, there will be high 2,3 BPG. It will be high 2,3 BPG. So must be increased CO2, decreased oxygen, low pH, high acidity, high hydrogen proton concentration, low OH, high 2,3 BPG, and that is the right answer, the last fourth one. Hemoglobin has how many subunits? We talked about that. It has 4 subunits, 2 alpha, 2 beta, 1 heme each, and 1 diatomic oxygen. 1 diatomic oxygen. How many subunits does myoglobin have? How many hemes? How many oxygens does it have? Myoglobin has 1 subunit, 1 heme, and 1 O2 each. Which form? The form is Fe plus 2, and that is known as the ferrous. The ferrous has longer letters, 7 letters, than the ferric that has less letters, yet it has more letters, 7 letters, ferrous, but it has less positive charges. These are plus 2. And that's the form that binds oxygen. Because remember, oxygen is diatomic, O2. So oxygen O2 binds to Fe plus 2. And ferric, Fe plus 3, does not bind oxygen. It is ferric, less letters, more pluses, does not bind oxygen. Ferrous, more letters, less pluses, 2 pluses, just like oxygen O2. Which amino acid stabilizes the bound O2 and acts as a gate for the ligand enter in both the subunits? Obviously, histidine. We talked about the involvement of histidine amino acid in the structure of hemoglobin. What is the relationship between positive feedback and hemoglobin? So the binding of the first oxygen favors the binding of the second, then the third. Cooperativity. Yes, cooperatively important as hemoglobin rapidly bypasses through the lung. Excellent. That's A. This one is the right answer. So binding of oxygen does not bind less. That's in the tissue. So the let go of the first oxygen will lead to let go of more oxygen. That's also a form of cooperativity. But in the lung, it's the more oxygen you add, the less T you're gonna get, the more R you're gonna become. Who is better for storing oxygen? Myoglobin is better for storing oxygen. Hemoglobin is better for delivering oxygen. We talked about that. The more effect, what happens is rapidly metabolizing tissues get more oxygen from hemoglobin. Rapidly metabolizing tissue get more oxygen from hemoglobin and more oxygen bound at the same partial pressure. Hemoglobin affinity for oxygen. Hemoglobin at the same pressure, less hemoglobin affinity for oxygen at the same pressure. So rapidly metabolizing tissue get more oxygen from hemoglobin and less oxygen is bound at the same pressure. Less oxygen are bound at the same pressure and less hemoglobin affinity for oxygen at the same pressure because hemoglobin need to let go of the oxygen to the tissue. What are the features of a rapidly metabolizing suffocating tissue? That will be both acid and carbon dioxide are released by rapidly metabolizing tissue. What is 2,3-BPG? 2,3-BPG is produced by exercising muscle cells due to rapid use of glycolysis. Glycolysis is when we break down glucose to give us ATP. And exercising muscle produce acid, carbon dioxide, and 2,3-BPG. And locks hemoglobin in the T state. Yes, locks it in the T state because it empties it from the oxygen. If it was carbon monoxide, it will leave it in the R state. It will make sure that it does not. So they always, these guys, these nasty guys, 2,3-BPG and CO2, they lock hemoglobin in the T state, emptying all that it has. That's why people who are smoking cigarette, they have 2,3-BPG, even in the lung where hemoglobin is supposed to get oxygen, 2,3-BPG is gonna lock it in the T state and prevent it from getting oxygen and becoming an R state. Hemoglobin function unloads oxygen in the lungs and loads carbon dioxide from the lungs. What is 2,3-BPG? What are the effects of smoking on the levels of 2,3-BPG? Carbon monoxide. What is that? What are the effects of carbon monoxide on hemoglobin? What are the effects of the acid, 2,3-BPG, and carbon monoxide on hemoglobin? Okay, so 2,3-BPG is a big concern for smokers. The blood of the smokers has high levels of 2,3-BPG, and it gets locked in the R state, passing through the, nope, it gets locked in the T state because they don't allow. An oxygen carrying capacity of the blood is reduced and carbon monoxide is higher. So this is the answer because it keeps it as a T. We just said that. It does not allow it to fill with oxygen. It keeps it empty from oxygen. What is the binding relationship between hemoglobin, carbon dioxide, and hydrogen protons? What is the binding relationship between hemoglobin, carbon dioxide,
and hydrogen bond and hydrogen protons, sorry. So these guys, so that question was describing in the setting of carbon monoxide. So in the carbon monoxide, carbon dioxide does not bind to heme nor do protons because carbon monoxide is preventing that. What usually happens is that carbon dioxide and protons bind to hemoglobin and they let go of the oxygen to the tissue. But in carbon monoxide is going to get between carbon dioxide and hydrogen protons and getting into hemoglobin and emptying it from oxygen, getting it from the R state to the T state. The carbon monoxide is going to lock hemoglobin in the R state, preventing carbon dioxide and the protons from binding to. So this answer, the question should have said, in the condition of carbon monoxide poisoning. What is the relationship between hemoglobin and carbon dioxide? They cannot bind to it. That means we cannot get oxygen out because if the oxygen gotta go out from hemoglobin only when these guys, carbon dioxide, 2,3-BPG, bind hemoglobin. But carbon monoxide is preventing their binding. So this question, I'm gonna give credit to everybody. The subunits for fetal hemoglobin are, yes, 2 alpha, 2 gamma. Put these protein enzymes in decreasing order of their respective affinity for oxygen, from the most affinity to the least affinity. So as the graph showed us, myoglobin has the most affinity. In the middle was fetal hemoglobin, and then to the rightmost, the least affinity, that is your adult hemoglobin. What is sickle cell anemia? Which amino acids are involved in the mutation? Are the sickle cells sickled all the time? No. They are only sickled when you need them the most, as your classmate so nicely said. When a patient has sickle cell anemia, you take blood sample from them, you look at it, they look biconcave, discoid, just like everybody else's red blood cell. It is only the problem, it is only when they are under stress and they need oxygen, then they start to sickle. There's a mutation from glutamic acid to valine. Red blood lose rounded shape and form sickle shape, and the change happens in low oxygen, like here. So this one is the answer. Mutation of valine to glutamic acid, and then red blood cells lose rounded shape and form sickle shape, and the change happens in low oxygen conditions when you need them the most. That's the answer. What is the graveyard of red blood cells? The spleen. And what is the fate? It is hemolysis. Lysis is breakdown. Hemo is blood, is breakdown of blood. That is the spleen hemolysis. That's the answer. The graveyard of blood cells is the spleen. What happens is hemolysis. What is sickle cell anemia? What we talked about that. We talked about that. Double that by mistake. People heterozygous for a mutated sickle cell anemia have a conferred resistance against malaria. Define an enzyme, the protein that speeds up the reaction by lowering the activation energy barrier. The substrate is the reactant, and the active site is the enzyme binding site for the catalyst. Yeah? Protein that speeds up the reaction by lowering the activation energy barrier. The substrate is the reactant. Yeah, that's the answer. A, protein that speeds up the reaction by lowering the activation energy barrier. The substrate is the reactant, and the active site is the enzyme binding site for catalysis. How does the enzyme speed up the reaction? How does the enzyme speed up the reaction? By lowering the activation energy. What is the activation energy of the reaction? How does the enzyme affect it? Are enzymes consumed in the reaction? Do enzymes alter the reaction and give different products? Do enzymes increase the rate of the reaction only in the forward direction, reverse direction, or both? So the answer should say that the enzyme lowers the activation energy barrier. The enzymes are not consumed in a reaction. The enzymes do not give different products in enzymes, and they increase the reaction rate both in forward and reverse direction. That's the right answer here, both forward and reverse. In the graph depicting enzyme catalyzed reaction rate as a function of time, where can I locate the transition state complex? The transition state complex is at the top. That is the activation energy is that long. Let me see if I can put what slide is. It's 118. Let me see if I can find it here. That is here. That's my saddle. That's my transition state complex. So the activation energy is the energy from the substrate up to the transition state complex, the saddle, the top point. The nadir is down here. That's the lowest point. That is where you have the product. But up here is where you have your transition state complex. What are the stop codon? You go away, you are away, you are gone. Which of the following is true regarding competitive and non-competitive inhibition? Both can overcome by increasing substrate. That's not true. Only competitive inhibitor when you add more substrate because it competes for the same site. So when you have more substrate, they're gonna overwhelm the competitive inhibitor. Competitive inhibition induces changes in the active site. It does not do that at all. It is the non-competitive inhibition has no effect on the enzyme affinity for substrate. Yes, because it binds to an allosteric, site, a site from behind. So that is 3 is the only answer. Enzyme kinetics regarding the Km and affinity, they have opposite values. So high Km meaning low affinity and low Km means high affinity. If you increase substrates in an enzyme catalyzed reaction, what happens? In the beginning, the reaction rate will increase initially until it reaches a certain point, then the reaction will not change, it will plateau, it will level off, will be the ceiling effect. What's an allosteric site? A site on the enzyme that is part of the competitive, nope, a site on the enzyme that does not bind the substrate, yeah, but control the enzyme is activated or inactivated. Yes, it's the back door. It's a site that's in the back that allows for the non-competitive inhibitor to bind, and that would regulate. It's allosteric. It's not substrate site, the allosteric. Intramolecularly, within the molecule, we can call isomerization of glucose 6-phosphate into fructose 6-phosphate. Rearrangement. Let's go look at that. Let's go look at Okay, glucose 6-phosphate. Look, 1, 2, 3, 4, 5, 6 carbons. 1, 2, 3, 4. 1, 2, 3, 4, 5, 6 carbons. If you count, they have the same number. They are just rearranged differently. So yes, it's a rearrangement. Yeah, it's true. Regarding the strands of DNA and RNA, DNA is double-stranded, has deoxyribose, no thymine. RNA is single-stranded, has uracil, and ribose sugar. Perfect, that's the answer. It is safe to say that ATP is nucleoside with 3 phosphates. Nucleotides are important for genetic information and energy storage and energy supply. So you store energy by putting phosphates on them. So you add adenosine monophosphate, you add one more phosphate, adenosine diphosphate, you add more phosphate, adenosine triphosphate. That's storing energy. Then we supply energy by removing the phosphates. So you go from adenine triphosphate to adenine diphosphate to adenine monophosphate. So the answer is A. DNA structure, phosphodiester bonds connect the 2 complementary strands together vertically, and hydrogen bonds connect the 2 complementary strands together horizontal. So the phosphodiester bonds connect nucleotides vertically within the same strand, and hydrogen bonds connect the 2 strands horizontally. Perfect. That's the right answer. Polarity of a DNA strand refers to the fact that the DNA lacks a hydroxyl group at one position of the ribose. No, that's deoxyribose. The two ends of the strand have the same chemical. No, they have different chemical groups. One has a phosphate. If the 5' end has a phosphate group, and the 3' end has an OH group. When two DNA pair, the two strands have different chemical groups. Yeah, the two ends of a strand have different chemical groups. The polarity of a DNA strand is indicated by referring to one end as the 3' and the other as the 5'. The 5' has a phosphate, 3' has an OH. Which structure on the 3' end and which structure in the 5' end? So the 5' end phosphate, 3' end hydroxyl. That's the right answer. What is meant that the DNA replication follows semi-conservative? Because the DNA is anti-parallel. Because the DNA is anti-parallel, we're going to have Okazaki fragment on the lagging strand. So for DNA replication, one completed strand contains only parental DNA, while the other contains only daughter. Yes, one strand contains one side that is parental, and the other side is daughter strand. After DNA replication, one completed strand contains a mixture of both parental DNA. No, it does not have a mixture. That's wrong. So after DNA replication, each strand contains one side that is parental, yep, and the other side that is daughter. Remember when I drew on the board that before cell division, we go into mitosis, we have the DNA, we cleave it in half, then we connect it, then we went from blue DNA that was cut in half into two copies, that blue with green, and then, you know, each strand, there are two strands, each strand is a parental strand that we chop it in half and we double it with a daughter strand. So each strand is made up of a parental original strand, known as the parental strand, and the second strand that we replicated is known as the daughter strand. Why must there be a lagging strand during the DNA synthesis? Because the DNA is anti-parallel. So you're going to make these Okazaki fragments. Why is it necessary that the DNA helicase unwind the DNA? It's important because it exposes the nitrogenous basis of the nucleic acid chain so that the new nucleotides can be added to create the daughter strand. What is the function of the topoisomerase? The topoisomerase cuts this very tightly coiled DNA, relieving the strain on the DNA ahead of the replication fork so we can begin the process of replicating. Which of the following is the first and the most important step in the polymerase chain reaction? We denature first, and then we add a primer, we exchange it, then we anneal, and we start making libraries. And we amplify the DNA. The polymerase chain reaction is used for constructing the wrapped maps. I'm going to give credit to everybody for this question. But the answer is amplifying all of the above, amplifying the gene of the interest and detecting the presence of these transcribed genes in new organisms. But this question, I'm going to give everybody credit for it because, you know, we haven't talked about the wrapped, but this is one of the advantages of the polymerase chain reaction. So these, all of the above, and everybody's going to get credit for that one. Where does eukaryotic and prokaryotic replication take place? Eukaryotic replication takes place in the nucleus, but the prokaryotic has no nucleus, so it's in the cytoplasm. In the cytoplasm. This is the answer, number 3. Leading strand of DNA being formed is continuous, but the lagging strand forms a number of small pieces. These are joined by a ligase that will stick together these small pieces known as the Okazaki fragments, ligases. Which of the following is a function of the telomerase? The telomerase is like plastic that is at the end of the shoelace. You know, your shoelaces, at the end of them, there's like a plastic piece. That plastic piece is protecting your DNA strand
from being deteriorated. It protects it from being deteriorated. And that protects the chromosomes from being deteriorating, because if the telomerase are all the way deteriorated and destroyed, that will initiate apoptosis and the cell is going to die. So the telomeres are there to protect the ends of the chromosome, like the shoelace has those plastics at the end to protect it from degradation. Telomerase is an enzyme that extends the telomeres, extends the telomeres. What kind of DNA damage is repaired using mismatch repair? That is a point mutation that is made during DNA replication. It was mismatch when the RNA matches with a G with a C, C with a G, T with an A with a U. So um, I'm sorry, that was for transcription. For replication, when the DNA polymerase is replicating a DNA strand, and it makes maybe one time, one in a million, makes a mistake. Instead of pairing up G with a C, it pairs up G with a T, then there will be a mismatch repair to repair that mismatch that happened. The mutation that causes sickle cell anemia is the result of a single nucleotide polymorphism from valine to glutamic acid. What kind of DNA damage is repaired using mismatch repair? Talked about that just now. Delete slide. Okazaki fragments are produced during the process of replication, the DNA. All the vitamins that are essential for blood clotting, it's known that vitamin K is essential to propagate the blood clotting. Plasmin is an enzyme in the blood plasma which causes the clotting of the blood by converting fibrinogen to fibrin. That's thrombin. Thrombin is the enzyme in the blood plasma that causes the clotting of the blood by converting fibrinogen to fibrin. But plasmin is the anticoagulant responsible for fibrin degradation during thrombolysis. So people who have hypercoagulability or they have a blood clot, they give them plasmin so it can lyse and break down that clot. So this is the opposite. Just replace plasmin with thrombin and thrombin with plasmin, and this statement would be correct. This statement is incorrect as of now. What prevents clotting of blood in the blood vessels? Heparin. Heparin ensures that it is an anticoagulant. Hemophilia is a genetic defect that prevents blood clotting, is a genetic mutation that makes coagulation happen. The genetic mutation in the gene that makes blood clotting factors. Yep. So the people with hemophilia, if they are injured, they can bleed to death. That's the answer is A, in the gene that makes the blood clotting factors. 2,3-BPG is a metabolic byproduct, toxic waste, and a stressful agent that causes hemoglobin to release unload bound oxygen. 2,3-BPG decreases the affinity of hemoglobin for oxygen. Yes, 2,3-BPG causes hemoglobin to switch from the R state full of oxygen to T state no oxygen. It's going to unload all of the oxygen to the tissue who's producing this toxic 2,3-BPG, and instead 2,3-BPG is going to bind to hemoglobin. What is a 2,3-BPG is not going to bind to the hemoglobin. It's just going to displace the oxygen. It is the carbon dioxide and hydrogen protons. They are the ones who bind to the hemoglobin. What is the amino acid that has an R group as an H and therefore is the only not chiral? That is your glycine. Disulfide bridges be formed between only the cysteines. What was the major conclusion that Christian Anfinsen got? That the proteins, the information dictating how the protein should fold is contained in the primary structure. Protein translated in the rough endoplasmic reticulum bound ribosomes attached to the rough endoplasmic reticulum are proteins that will be used, exported out, not locally, be sent out. And these the proteins who are synthesized in the free-floating ribosomes, they will be used inside. And the ones who are made in the Ribosomes attached to the rough endoplasmic reticulum. They will be packaged in the Golgi body, and they will be exported for use outside of the cell, or they will be inserted in the lysosome. So this is incorrect. Replace the proteins translate. So replace the function. So just switch them around. It is the proteins who are synthesized, translated, or synthesized by ribosomes attached to the rough endoplasmic reticulum. They will be shipped out, sent out, exported out of the cell, or sent to the Golgi body or the lysosome. But it is the proteins who are made by by free floating ribosomes, these are the proteins who will be used locally inside the cell. Salting in refers to the effect where increasing the ionic strength of a solution decreases the solubility. So that is salting out. So salting in increase the solubility. Salting out decrease the solubility, and that's going to crystallize our protein of interest. 136, so it's false. 136 is false. Yeah, it's false. Salting refers to the effect where increased ionic strength of a solution decrease the solubility. No, that's the sorting out. What type of interaction happens in affinity? So we're going to have a non-covalent. So that's a bond, like when you have nucleic acids attached to each other, like in the double helix, G with a C, A with a T. You know that kind of bond is a dashed bond. That's a non-covalent hydrogen bond. Ion exchange chromatography does not depend on which of the following. Ion exchange depends on electrolytes, depends on the pH, the salt concentration, the buffer. It does not depend on the weight. That is the gel chromatography. Dash is a liquid column chromatographic method of separating solute molecules according to differences in molecular size. That's the gel filtration chromatography. Okay, it's 12:30 a.m. Thank you and best of luck.